The human body did not evolve in kitchens. It evolved in uncertainty. For nearly all of our existence, food was rare, unpredictable, sometimes absent for days. The metabolism we carry today was shaped by scarcity, not abundance. Yet we now eat from sunrise to midnight, as if constant fuel were a biological right. Quietly, without drama, we may be living in a way our cells have never truly known. If hunger once helped keep us alive, what happens when it disappears completely? And why was the human body never designed for constant food? For most of the 200,000 years that our species has existed, the human body moved across landscapes where certainty was rare. Small groups walked through dry grasslands, river valleys, and open woodland, carrying little and depending on what could be found or caught. Food did not wait in storage. It appeared in seasons, in migrations, in brief windows of opportunity. A successful hunt might provide abundance for a short time. Days afterward could pass with almost nothing. In that world, the metabolism that still operates inside us began to take shape. It was not built around steady input. It was shaped by irregular arrival. When we ask why the human body was never designed for constant food, we must begin with that environment, because physiology does not emerge from comfort. It emerges from pressure. The earliest members of Homo sapiens did not wake to stocked kitchens. They woke to uncertainty. Archaeological evidence from sites in East Africa shows repeated movement patterns rather than permanent food storage. Animal bones with cut marks indicate episodic access to meat. Plant remains suggest gathering that depended heavily on season and climate. There is no sign of daily predictability. Instead, there is variability. Over tens of thousands of years, that variability became the background against which natural selection operated. Bodies that could endure gaps between meals survived more often than bodies that required constant replenishment. In such conditions, flexibility was more valuable than fullness. The human liver learned to release stored glucose when intake paused. Fat tissue became not merely padding but insurance. Hormonal systems evolved to shift between using immediate fuel and stored reserves. None of these adjustments required a steady stream of calories. They required adaptability. Even today, when a person has not eaten for several hours, the body does not collapse. It adjusts. That adjustment is not an emergency response invented by modern science. It is a deep inheritance from a time when hours without food were ordinary. Around 300,000 years ago, in fluctuating African climates, rainfall patterns shifted repeatedly. Lakes expanded and contracted. Herd animals moved according to water and vegetation. Human groups followed. Each movement meant that food access changed. There were days of success and days of waiting. The metabolism that survived such cycles was one capable of switching fuel sources. Glucose from recent meals could power immediate activity. Fat stored over weeks could sustain longer gaps. This capacity to move between fuels is not a side feature. It is central to how the human body was formed. When food is constant, that switching becomes less necessary. But in the ancestral world, it was essential. The archaeological record also shows limited evidence of long-term storage technologies until very late in prehistory. Without pottery, refrigeration, or agriculture, surplus could not easily be preserved. What was hunted or gathered was consumed quickly or shared. The absence of storage reinforced the rhythm of irregular intake. A body designed for constant food would have been fragile in such an environment. It would have depended on conditions that did not exist. Instead, natural selection favored metabolic resilience, the capacity to function while hungry, to think clearly enough to track prey, to walk long distances on limited intake, became part of the species. When we examine our current environment of endless availability, we see something profoundly different from that original setting. Supermarkets, processed foods, and artificial light extend access beyond daylight and season. Yet the biological systems inside us were tuned in landscapes where nothing was guaranteed. The question is not whether constant food is comfortable. The question is whether comfort aligns with the pressures that shaped our design. To understand why the human body was never designed for constant food, we must first accept that it was designed in a world where constancy rarely existed at all. In that early world of shifting climate and uncertain supply, survival did not depend on eating often. It depended on enduring interruption. When we imagine the daily life of early Homo sapiens, it is tempting to picture steady activity supported by steady meals. Yet the archaeological and ethnographic records suggest something quieter and less predictable. A group might succeed in bringing down a large animal, consuming significant calories within a short span. Then, for several days, intake could fall sharply. The body that endured was not the one that required balance each day, but the one that tolerated imbalance without immediate breakdown. This pattern was not occasional. Over thousands of generations it became normal. Around 200,000 years ago, human populations were small and widely dispersed. Food was acquired through foraging, hunting, fishing, and opportunistic gathering. Seasonal fruits appeared briefly and vanished. Tubers required effort to extract. Herds migrated beyond reach. Even coastal groups experienced fluctuation depending on tides and weather. These conditions created a metabolic rhythm shaped by availability rather than preference. The body learned to anticipate uncertainty. Hormonal systems regulating appetite, energy use, and storage became sensitive to scarcity. When intake dropped, the body reduced non-essential expenditure. Movement remained possible, but energy was allocated carefully. The brain, which consumes a large proportion of available glucose, relied on mechanisms that allowed it to function even when meals were absent for extended hours. This capacity to remain cognitively active during hunger would have been essential for tracking animals or navigating terrain. Without it, decision-making would falter at precisely the moment when clarity was most needed. From an evolutionary perspective, traits that allowed individuals to operate during gaps between meals increased reproductive success. Over time, such traits became common. This is not speculation but inference drawn from comparative physiology and anthropological observation of recent hunter-gatherer societies. Groups studied in arid regions of Africa and Australia have shown irregular eating patterns shaped by environment rather than by clock time. While these communities are modern and influenced by contact, their subsistence strategies still reflect principles of fluctuation. Meals are often dependent on successful procurement rather than on fixed schedules. This suggests that the human body retains compatibility with intermittent intake because that compatibility was forged over vast stretches of pre-agricultural history. Even during the later Paleolithic period, when tool use became more refined, predictability did not dramatically increase. Improved weapons raised hunting success rates, but they did not eliminate environmental variation. Droughts, cold spells, and migrations continued to influence access. There was no infrastructure to guarantee daily abundance. Storage technologies remained limited. The absence of preservation meant that excess food could not always be saved for lean days. As a result, feast and pause existed side by side. This rhythm influenced not only metabolism but social structure. Sharing became essential because surplus could not be hoarded indefinitely. Cooperation buffered scarcity, but it did not erase it. In such a setting, constant food would not merely have been unnecessary. It would have been unrealistic. The human digestive system, endocrine signaling, and energy storage patterns emerged within this dynamic landscape. When we consider modern habits of continuous eating, we are observing behavior made possible by recent technology, not by ancient design. The body we inhabit was stabilized by repetition across generations, but that repetition involved cycles of access and absence. The deeper we look into the environmental history of our species, the clearer it becomes that regular interruption was part of the original pattern. The idea of uninterrupted intake belongs to a different era. To understand why the human body was never designed for constant food, we must continue tracing how those early cycles became embedded in our biology. Across tens of thousands of years, repetition carved expectation into biology. When an organism repeatedly encounters a certain pattern, natural selection does not merely tolerate that pattern, it builds around it. For early humans, the repeating pattern was not constant nourishment, but alternating access. Days of movement under open sky were not punctuated by predictable meals at fixed hours. They were shaped by opportunity. A successful hunt might mean eating until satisfied. A failed attempt meant conserving strength and waiting. This alternation did not occur once or twice in a lifetime. It unfolded across countless generations. Over roughly 200,000 years of Homo sapiens history, the majority of that time passed without agriculture, without storage, without artificial preservation. The body that endured was one that could shift smoothly between intake and absence without damage. Around 100,000 years ago, human groups expanded into new territories, including colder regions. In such climates, food availability could become even more irregular. Animal migrations determined opportunity. Harsh winters reduced plant options. Archaeological sites in Eurasia reveal evidence of episodic hunting success followed by leaner phases. Bone assemblages often show concentrated events of butchery rather than evenly distributed remains that would suggest daily predictability. These patterns imply bursts of availability rather than constant supply. Under such conditions, metabolic flexibility became more than useful. It became necessary. The human body developed the capacity to store surplus energy efficiently in adipose tissue, not as an error but as preparation. Fat represented stored time. It allowed an individual to move, think, and survive during days when no new calories arrived. The liver's ability to release glucose, the muscle's capacity to draw on internal reserves, and the hormonal coordination that shifts energy use were refined in this long context of uncertainty. These processes are quiet and automatic. They do not require conscious decision. They are reminders that the body expects fluctuation. If it had been designed for constant food, its systems would fail quickly during interruption. Instead, interruption is tolerated. In fact, brief absence of intake triggers organized internal responses rather than collapse. When intake pauses, insulin levels fall. Stored energy becomes accessible. Appetite signals rise and fall in cycles. None of this reflects malfunction. It reflects design shaped by irregularity. Even the human brain, often described as energy-dependent, contains mechanisms allowing it to adapt to varying fuel availability. This resilience would have been essential when groups tracked animals across long distances without guarantee of immediate reward. A mind that could function only when freshly fed would have reduced survival chances. The persistence of these traits suggests that for most of human existence, gaps between meals were not rare exceptions. They were expected events. It is only within the last few centuries, and especially within the last few decades, that continuous access to calorie-dense food has become widespread in many societies. Compared to the deep timeline of human evolution, this change is extremely recent. The body has not undergone fundamental redesign in this short interval. It still carries assumptions forged in landscapes where constancy did not exist. When we examine modern habits of eating from early morning until late at night, often without extended pauses, we are observing behavior that the ancestral environment never required. The question, then, is not whether we can live with constant food. Clearly we can. The question is whether our internal systems were ever structured around that condition. As we continue to move forward, the focus shifts from the landscape outside the body to the signals within it, because the environment shaped the design, but the design now operates quietly inside each of us, still tuned to cycles rather than continuity. If the early landscape shaped the human body through uncertainty, then hunger itself must be understood not as a defect, but as a signal that emerged from that environment. In modern settings, hunger is often interpreted as something to eliminate quickly. It feels uncomfortable, distracting, even alarming. Yet for most of human existence, that sensation was not an emergency. It was information. It guided movement, attention, and effort. To understand why the human body was never designed for constant food, we must examine hunger as a biological message rather than a failure of the system. Deep within the brain, regions that regulate appetite respond to changing levels of energy in the bloodstream. Hormones such as ghrelin rise before anticipated intake, while insulin and other signals shift after food is consumed. These patterns did not arise in a world of uninterrupted supply. They developed in a world where intake fluctuated. Hunger signaled that stored energy should begin to be mobilized, that attention should turn towards seeking resources. It was a cue aligned with survival behavior. 200,000 years ago, when small bands moved across African savannas, hunger would have sharpened awareness rather than paralyzed it. Ethnographic observations of more recent foraging communities suggest that individuals often function actively even when meals are not immediate. The body compensates. Blood glucose is maintained within a relatively narrow range through internal regulation. The liver releases stored glycogen. fat tissue begins to provide energy substrates. These processes unfold gradually, not catastrophically. They reveal that the body expects intervals between meals. If constant food had been the default condition during evolution, such intricate regulatory systems would not have been necessary. Instead, we see a network designed to handle absence calmly. Even the cyclical nature of appetite hints at this design. Hunger does not rise endlessly in a straight line. It tends to appear in waves, often influenced by habitual timing, stress, and activity. This wave-like pattern suggests flexibility rather than dependence. The brain and endocrine system coordinate to maintain function during gaps. Cognitive performance does not instantly collapse when intake pauses for several hours. Historically, this resilience would have allowed hunters to remain focused during long pursuits and gatherers to travel between dispersed food sources. The ability to operate without immediate replenishment increased survival odds in unpredictable environments. From a biological perspective, hunger serves as a bridge between stored energy and behavior. It encourages action without implying crisis. When food becomes constant, the signal still exists, but its context changes. Instead of guiding survival activity in a variable landscape, it may prompt consumption in an environment already abundant. The internal design remains anchored to ancient conditions. Around 100,000 years ago, repeated cycles of scarcity reinforced these signaling pathways. Individuals whose bodies could regulate appetite without destabilizing blood sugar had advantages. Over generations, such regulation became typical of the species. What we carry today is the result of that long filtering process. The discomfort of hunger, when mild and temporary, is part of a system built to tolerate fluctuation. It was never meant to be eliminated entirely from daily experience. To see hunger only as a problem is to overlook its evolutionary role. As we move deeper into the biology of energy use, the focus shifts from the signal itself to what the body does when that signal persists. Because the presence of hunger is only the beginning of a coordinated internal response, shaped long before constant food became possible. When hunger continues beyond its first gentle wave, the body does not enter chaos. It enters adjustment. This adjustment is not dramatic, and in most healthy individuals it unfolds without conscious awareness. The endocrine system begins to shift priorities. Insulin levels decline as incoming glucose from food is no longer present. In response, stored glycogen in the liver is gradually broken down to maintain blood sugar within a narrow, stable range. This stability is essential because the brain relies heavily on glucose for immediate energy. The fact that blood sugar can remain regulated for many hours without new intake is not an accident. It reflects a long evolutionary history in which gaps between meals were expected rather than rare. Roughly 200,000 years ago, when Homo sapiens populations were adapting to fluctuating African climates, the ability to preserve cognitive clarity during periods without food would have carried significant survival value. A hunter tracking animals across open terrain could not afford confusion or weakness simply because a meal had not yet occurred. The body therefore developed layers of redundancy. After glycogen stores begin to diminish, metabolic pathways shift again. Fat tissue, accumulated during previous periods of abundance, becomes increasingly important. Fatty acids are released into the bloodstream and used by muscles and other tissues as fuel. Over time, the liver can convert some of these fatty acids into ketone bodies, which the brain can also use as an alternative energy source. This capacity does not imply that prolonged starvation is harmless. Severe deprivation is dangerous, but short to moderate intervals without food are well within the range that the human body evolved to manage. The existence of these metabolic transitions demonstrates that constant intake was not assumed in the original design. If the ancestral environment had provided uninterrupted nourishment, there would have been little selective pressure to maintain such flexible systems. The repeated exposure to feast and scarcity strengthened them instead. Archaeological and anthropological evidence reinforces this interpretation. Isotopic analysis of ancient bones suggests variation in diet composition across seasons. Cut marks on animal remains appear clustered rather than evenly spaced across time, implying episodic access to meat. These patterns support the view that energy intake was irregular. Within such a framework, the body's capacity to transition smoothly between fuel sources becomes logical rather than exceptional. It was an adaptive necessity. Even at the level of cellular signaling, Enzymes involved in fat oxidation and glucose production respond dynamically to changing intake. This responsiveness is not passive. It is active regulation shaped over thousands of generations. Around 100,000 years ago, as human groups migrated into new ecological zones, metabolic flexibility would have remained advantageous. In colder regions with reduced plant diversity, successful hunting events might have been separated by days. In coastal environments, tides and weather influenced access to seafood. In each setting, the body relied on stored reserves between opportunities. These recurring experiences left their imprint on physiology. When food is now available every hour of the day, the same regulatory systems continue to operate, but the pattern they evolved for has shifted. Hunger may arise not because reserves are depleted, but because cues linked to routine, emotion, or environment trigger the familiar signal. The biological machinery remains tuned to cycles of intake and pause. Understanding this helps clarify why the human body was never designed for constant food. It was designed to interpret absence as manageable and to respond with coordinated adaptation. As we continue, attention turns to how energy storage itself became a central feature of survival, because the ability to endure hunger depended not only on signaling but also on what the body chose to preserve during times of plenty. If hunger is a signal and metabolic adjustment is the response, then the final piece of this pattern is memory. Not memory in the conscious sense, but biological memory, shaped by repetition across generations. The human body does not simply react to the absence of food in the present moment. It carries a long history of exposure to absence. That history has influenced how energy is prioritized, how appetite is regulated, and how survival mechanisms are balanced against long-term growth. When we ask why the human body was never designed for constant food, we are asking whether uninterrupted intake aligns with the expectations written into that biological memory. Around 200,000 years ago, small human populations survived by navigating unpredictable ecologies. In those environments, individuals who conserved energy efficiently during lean periods were more likely to live long enough to reproduce. This conservation was subtle. When intake fell, non-essential processes could slow slightly. Physical movement remained possible, but internal expenditure was carefully allocated. The body favored functions critical for immediate survival. Over time, natural selection preserved regulatory systems that supported this flexible conservation. Appetite hormones adjusted in cycles, stress responses coordinated with resource availability. Energy storage was protected when scarcity loomed. None of these traits required constant feeding. In fact, their value emerged precisely because feeding was not constant. Even today, when a healthy adult misses a meal, the body does not interpret it as catastrophic. Glycogen supplies buffer the gap. Fat reserves extend endurance. Hormonal changes occur gradually rather than abruptly. This layered resilience suggests that intermittent intake is within the normal operating range of human biology. The discomfort that may accompany mild hunger does not necessarily indicate damage. It reflects signaling calibrated in a world where brief gaps were routine. Ethnographic studies of traditional foraging groups in arid regions show that meal timing often reflects opportunity rather than schedule. Although modern influences complicate direct comparison, these observations support the idea that irregular intake remains compatible with human physiology. The persistence of these adaptive responses highlights a key point. Evolution does not optimize organisms for every possible future environment. It shapes them for the conditions repeatedly encountered. For Homo sapiens, those conditions involved cycles of access and absence extending across vast stretches of time. The agricultural transition, which began roughly 10 to 12,000 years ago, introduced more predictable food supplies in certain regions. Yet even then, harvest cycles, storage limitations, and periodic famine preserved elements of fluctuation. Only in the last century, and especially in the last few decades, has large-scale constant availability become widespread in many societies. Compared to the deep timeline of human evolution, this shift is extremely recent. The internal regulatory systems shaped over hundreds of thousands of years have not been fundamentally redesigned in that brief interval. They continue to operate as if variation is normal. When intake becomes continuous, these systems may still respond with the same hormonal patterns and storage tendencies developed under scarcity. This does not imply that modern abundance is inherently harmful. It suggests that it differs from the context in which our metabolism was formed. The body expects intervals of pause. It recognizes hunger as a cue, not as an error. It activates stored energy when intake stops. These responses are not relics. They are active features of present-day physiology. Understanding hunger as part of a long adaptive rhythm reframes the central question. If the human organism evolved to function across cycles of feeding and fasting, what does it mean to live in an environment where that cycle is rarely allowed to unfold? If hunger is tolerated and metabolic switching is possible, then we must look at the structure that makes endurance practical. That structure is energy storage. In modern conversation, body fat is often described as excess, as something accidental or undesirable. Yet in the long arc of human evolution, stored fat was not a mistake, it was protection. To understand why the human body was never designed for constant food, we must recognize that it was designed to survive when food stopped arriving, and survival during absence depends on what was saved during presence. Around 200,000 years ago, early Homo sapiens lived in environments where success was uneven. A hunting expedition might yield a large animal, providing substantial calories for a short period. During such moments, eating beyond immediate need was not indulgence. It was preparation. The body responded by converting surplus energy into stored triglycerides within adipose tissue. This conversion required coordination between hormones, enzymes, and cellular transport systems. Over thousands of generations, individuals whose bodies could efficiently store energy during abundance had an advantage when scarcity returned. Those who stored poorly were more vulnerable during lean days. Natural selection does not reward comfort. It rewards survival through variation. Fat tissue, therefore, became a metabolic reserve that extended time. It allowed muscles to keep working and the brain to remain alert when external intake paused. The presence of fat storage capacity across human populations suggests that repeated exposure to fluctuating supply shaped this trait deeply. Even today, when caloric intake exceeds immediate expenditure, the body tends to store the difference. This tendency is sometimes framed as a flaw in modern contexts. From an evolutionary perspective, it reflects continuity with ancestral conditions. The environment that shaped our physiology did not offer predictable three-meal patterns separated by stable refrigeration. It offered irregular bursts of opportunity. Under such conditions, rapid storage was adaptive. Archaeological evidence supports the idea that food access was episodic rather than constant. Bone remains at Paleolithic sites often indicate concentrated processing events, suggesting that when animals were successfully hunted, groups consumed large amounts within limited windows. There is little evidence of long-term preservation during most of prehistory. Without reliable storage technology, the safest place to keep energy was within the body itself. Adipose tissue functioned as a portable storehouse. Around 100,000 years ago, as human groups expanded into colder climates, the importance of insulation and stored fuel may have increased further. Subcutaneous fat provided not only energy but also thermal buffering. Individuals with sufficient reserves were better equipped to endure environmental stress. This dual role of fat underscores that it was not a passive accumulation. It was integrated into survival strategy. In an environment of constant food, such aggressive storage may appear excessive. In a landscape defined by cycles, it was prudent. The human endocrine system evolved to favor storage when surplus appeared, because surplus could not be assumed to return quickly. When food now remains available at all hours, the same biological impulse persists. The body continues to interpret abundance as temporary unless proven otherwise. This is not a conscious calculation. It is an inherited bias shaped by repeated scarcity across deep time. Understanding energy storage as an adaptive feature shifts how we interpret modern eating patterns. The question is not why the body stores. The question is why it would have evolved not to store, given the conditions of its origin. As we continue, attention turns to how this storage interacts with modern patterns of uninterrupted intake, because the design that once protected survival now operates within an environment it did not anticipate. Energy storage in the human body is not random accumulation. It follows a coordinated biological pattern shaped over long stretches of evolutionary time. When food becomes available after a period of relative scarcity, hormonal signals shift rapidly. Insulin rises, guiding glucose into cells and encouraging the conversion of excess energy into fat. This process is efficient by design. Efficiency in storage would have increased survival when the next successful hunt or harvest was uncertain. Around 200,000 years ago, individuals who could transform surplus calories into stable reserves were more likely to endure the next dry season or failed expedition. The ability to store was not merely helpful, it was protective. Adipose tissue is metabolically active, not inert. It communicates with the brain and other organs through hormones such as leptin, which signals the presence of stored energy. In ancestral environments, these signals helped regulate appetite and expenditure in relation to available reserves. When stores were sufficient, the body could tolerate temporary absence of intake without initiating crisis responses. When stores were depleted, hunger signals intensified, guiding behavior toward renewed foraging. This feedback loop developed within a landscape defined by irregular supply. The loop assumes fluctuation. It does not assume constant replenishment. Archaeological findings reinforce the idea that surplus, when it appeared, was often consumed in concentrated intervals. Evidence from Paleolithic sites suggests episodic access to large game. Without reliable preservation methods, energy not immediately used had to be stored biologically. The body became the storage site. Over generations, metabolic pathways favoring rapid fat synthesis after abundance were retained. Those pathways remain active today, even in modern societies with stable food access. The body responds to caloric excess with storage because it interprets abundance through ancient assumptions. Roughly 100,000 years ago, as human groups migrated into varied climates, these storage mechanisms likely became even more significant. In colder regions, higher energy expenditure combined with unpredictable hunting success increased the value of reserves. Fat provided both insulation and fuel. Individuals lacking sufficient storage capacity would have faced greater risk during prolonged cold or food shortage. The persistence of adipose tissue as a central energy reserve across human populations indicates that fluctuating intake was not a rare anomaly. It was common enough to influence biological design. When constant food becomes available, the same storage system continues to operate. However, the rhythm changes. Instead of storing in preparation for expected scarcity, the body may store without ever experiencing the absence that once balanced accumulation. The endocrine signals do not fundamentally distinguish between ancient surplus and modern abundance. They respond to energy presence. This does not mean the body is malfunctioning. It means it is operating under rules established in a different ecological context. The human metabolism expects cycles of storage followed by usage. When usage does not follow, reserves may gradually increase. From an evolutionary standpoint, this is not irrational. It reflects preparedness. Understanding this helps clarify the central question. The body was designed to capture and preserve energy when possible because food was not constant. If food had been reliably available each day across evolutionary time, such aggressive storage would have offered little advantage and might have been reduced through selection. Instead, it was reinforced. The design of human adipose tissue, hormonal signaling, and metabolic switching reveals a system prepared for interruption rather than continuity. As we move forward, the focus shifts toward how modern patterns of uninterrupted intake alter the balance between storage and release. Because the design remains cyclical even when the environment becomes steady. The presence of stored energy only makes sense within a system that expects it to be used. Fat tissue was not shaped to expand without limit, it was shaped to buffer time. In an ancestral environment where intake rose and fell unpredictably, storage and release were paired processes. Surplus entered the body during moments of success. Gradually, during days of reduced access, that surplus was drawn down. The cycle completed itself. When we ask why the human body was never designed for constant food, we are also asking why storage mechanisms evolved with an assumption of future scarcity. The answer lies in repetition across deep time. For roughly 95% of human existence, agriculture did not exist. There were no grain silos, no refrigeration, no global transport of calories across seasons. Energy that could not be preserved externally had to be preserved internally. This meant that after a successful hunt or gathering event, the body shifted into conservation mode. Insulin guided nutrients into tissues. Fat cells expanded slightly, preparing for interruption. When interruption came, the reverse occurred. Hormonal signals shifted. Lipolysis began, releasing fatty acids into circulation. Muscles and organs drew from these reserves. This alternating pattern reinforced balance. The storage system was not isolated from the release system. They evolved together. Around 150,000 years ago, as populations spread across varied ecosystems, from open savannas to wooded regions, the unpredictability of food sources remained a constant theme. Even coastal groups who had access to shellfish and fish were subject to tides, storms, and seasonal change. Archaeological layers often show pulses of resource use rather than smooth continuity. Such pulses would have reinforced the metabolic expectation that abundance is temporary. The body does not assume that excess today guarantees excess tomorrow. It prepares. In modern contexts, where food can be accessed repeatedly throughout the day and across all seasons, the release phase of this ancient cycle may be shortened or absent. The storage phase continues because the signals that initiate it remain intact. This asymmetry between storage and release is not the result of a new biological design. It is the result of an environment that no longer mirrors the one in which the design emerged. The endocrine system still responds to caloric surplus with efficiency. It still protects energy reserves when intake pauses, even briefly. What has changed is the frequency and duration of those pauses. In ancestral settings, pauses were long enough and common enough to balance accumulation. In many modern settings, pauses are minimal. The biological system has not recalibrated over the few thousand years since agriculture began, and certainly not over the last century of industrial food production. Evolution operates across far longer timelines. This does not imply inevitability or determinism. It suggests context. Fat storage was never meant to operate in isolation from periodic use. It was part of a broader rhythm that included movement, effort, and intervals without intake. The body that carries energy efficiently is the same body that expects to draw upon that energy. When constant food becomes the norm, the expectation embedded in metabolism does not disappear. It remains quietly aligned with cycles that shaped it. To understand why the human body was never designed for constant food, we must see storage not as excess alone, but as half of a paired system built for fluctuation. The next step is to examine the physical evidence left behind by those fluctuations, because bones, tools, and settlement patterns still carry traces of how irregular intake once structured human life. Long before written records, long before agriculture reshaped landscapes, human lives left quieter traces. Stone tools, fragmented bones, scattered hearths, and layers of sediment became the evidence from which we reconstruct daily rhythms. When we examine these traces carefully, a consistent pattern appears. Food access was uneven. The archaeological record does not show steady, uniform consumption across time. It shows pulses. These pulses help answer why the human body was never designed for constant food, because they reveal the ecological reality within which our physiology formed. At many Paleolithic sites across Africa and Eurasia, animal remains are found in concentrated deposits. Cut marks on bones indicate coordinated butchery events. The clustering of such remains suggests moments of successful hunting rather than daily predictable intake. A large animal, once obtained, could provide substantial calories for a small group, but it could not be preserved for extended periods without advanced storage technology. Much of it would have been consumed within a short window. After that window closed, the group returned to uncertainty. The archaeological layers do not reflect evenly spaced access to meat across days and weeks. They reflect episodes separated by gaps. This episodic pattern mirrors the feast and absence cycle that shaped metabolic flexibility. Plant remains tell a similar story. Evidence of gathered seeds, fruits, and tubers appears seasonally in certain regions. Charred plant fragments in hearth sites indicate consumption linked to availability rather than to a fixed schedule. In savanna and woodland environments, fruiting seasons were brief. Tubers required labor to extract and were not always abundant. The variability in botanical remains across strata suggests shifting reliance depending on what the environment offered at a given time. The human body that evolved under such conditions did not encounter constant intake. It encountered variability structured by climate and ecology. Around 100,000 years ago, as Homo sapiens expanded into the Levant and later into Europe and Asia, this variability persisted. In colder regions, large herbivores became crucial resources, yet migration patterns of these animals introduced unpredictability. Archaeological evidence from caves and open-air sites shows repeated occupation layers separated by periods of absence. Human groups moved when resources shifted. This mobility itself implies that food was not reliably available in one place. If constant supply had existed, permanent settlement would have appeared far earlier in human history. Instead, for most of our existence, mobility remained adaptive. Mobility and irregular intake are closely linked. The absence of large-scale storage technologies before the Neolithic transition reinforces this interpretation. Without pottery or durable containers, surplus could not be safely preserved across long intervals. The safest storage remained biological, within the body. Adipose tissue functioned as a living reserve because the environment did not offer external stability. Archaeology, therefore, aligns with physiology. The physical record of human subsistence supports the view that food access came in waves. This matters because biological design reflects repeated exposure, not isolated events. If for tens of thousands of years intake rose and fell according to ecological cycles, then metabolic systems would adapt accordingly. Constant food would have been an anomaly. The stones and bones left behind do not tell us how individuals felt hunger. They tell us something equally important. They show that the context in which hunger occurred was one of structural unpredictability. As we move further, we must look more closely at how these archaeological patterns intersect with population structure and generational turnover, because the design of metabolism was filtered not only by environment, but also by who survived within it. Archaeology does not speak in sentences, but in distributions. When layers of occupation are studied over time, what emerges is not a picture of uniform daily intake, but one of variability shaped by movement, climate, and opportunity. At many Paleolithic sites, researchers observe alternating phases of dense activity and near absence. Hearths appear in clusters. Stone tools are concentrated in specific layers. Animal bones show intensive processing in particular moments rather than continuous accumulation. These patterns imply that food acquisition was tied to events, not to steady repetition. Such evidence deepens the argument that the human body was never designed for constant food, because the environment that shaped it did not provide such constancy. In regions of East Africa considered central to early Homo sapiens evolution, faunal remains often reflect opportunistic hunting strategies. Smaller animals, large ungulates, and even scavenged carcasses appear in assemblages that suggest flexibility rather than predictability. The variability in prey species across nearby sites indicates that humans adapted to what was available at the time. If one source diminished, another was pursued. This flexibility in subsistence would have required metabolic systems capable of adjusting to fluctuating caloric and macronutrient intake. A body expecting uniform daily meals would have struggled in such settings. Instead, the archaeological diversity of prey and plant remains aligns with biological adaptability. Around 70 to 100,000 years ago, as humans expanded beyond Africa, this pattern continued. In the Levant, cave sites reveal periods of occupation corresponding with favorable climatic phases. During colder or drier intervals, occupation thins or disappears. These shifts mirror ecological change. Where humans moved, food followed environmental constraints. There is little indication of year-round abundance within a single location over extended spans. Even in resource-rich areas, seasonal patterns structured availability. The repeated need to relocate reinforces the idea that food supply was not stable enough to support constant intake without interruption. The archaeological absence of large storage pits, permanent granaries, or preserved food caches in most Paleolithic contexts further strengthens this interpretation. Unlike later agricultural societies, early foraging groups lacked infrastructure to buffer against ecological fluctuation on a large scale. Without long-term storage, intake necessarily mirrored opportunity. When resources were present, consumption rose. When resources were scarce, intake declined. This oscillation was not rare. It was embedded in daily and seasonal life. Over many generations, Individuals whose metabolism tolerated this oscillation without severe instability were more likely to survive and reproduce. Thus, archaeology and evolutionary biology converge. The physical remnants of past life reveal episodic procurement and mobility. The physiological traits observed in modern humans reveal tolerance for gaps between meals and efficient storage during abundance. These two lines of evidence reinforce each other. They suggest that the design of human metabolism is coherent with a subsistence pattern defined by cycles rather than constancy. As we continue, attention shifts toward how population size, life expectancy, and generational turnover influence which metabolic traits were preserved, because the archaeological record shows us patterns of behavior, but survival patterns determined which bodies carried those adaptations forward. Beyond tools and bones, archaeology also reveals something about scale. For most of human history, populations were small, scattered, and vulnerable to environmental pressure. When a group depended on wild plants and animals, the success of one season could not guarantee the next. Sediment cores and paleoclimate data show repeated shifts in rainfall and temperature across Africa and Eurasia during the last 200,000 years. Lakes expanded and contracted, grasslands alternated with woodland, herd distributions changed accordingly. Human subsistence followed these shifts. The material record reflects adaptation to instability rather than life within steady abundance. This broader environmental evidence strengthens the claim that the human body was never designed for constant food, because constancy rarely existed at the ecological level. In many regions, archaeologists find repeated cycles of occupation and abandonment. A cave might show evidence of intensive use during a favorable climate window, then silence for centuries. Such patterns indicate that food availability was tied to environmental fluctuation. When resources diminished, people moved. Mobility was not optional, it was essential. Mobility implies variability in intake. A community traveling across terrain in search of water or migrating herds would not have maintained fixed daily meal schedules. Instead, food acquisition would have depended on encounter and success. This pattern recurs across continents and time periods. Even coastal settlements, often considered more stable due to access to marine resources, display seasonal signatures. Shell middens accumulate heavily during particular times of year, suggesting focused harvesting when tides and breeding cycles aligned. These deposits are rarely evenly distributed across all seasons. Such evidence implies pulses of abundance separated by quieter intervals. The rhythm is visible in the debris itself. Around 40 to 60,000 years ago, as human populations expanded further into Europe and Asia, climatic instability intensified during glacial cycles. Cold periods reduced plant diversity and altered animal migration routes. Archaeological layers from this era often reveal abrupt changes in tool types and prey selection, indicating adaptive responses to shifting food sources. These shifts underscore the lack of long-term dietary predictability. The body evolving under these conditions would have benefited from mechanisms that smoothed energy supply internally when the external world offered no guarantees. The absence of durable storage infrastructure during this vast period remains significant. Before the development of agriculture roughly 10 to 12,000 years ago, there is limited evidence of large-scale food preservation. Without granaries, fermentation vats, or refrigeration, communities could not insulate themselves fully from ecological cycles. Surplus, when obtained, was fleeting. Biological storage filled that gap. This is where archaeology meets physiology once again. The scattered remnants of meals, the episodic burn layers of hearths, and the intermittent occupation of sites collectively describe a subsistence system structured by fluctuation. Over generations, only those individuals whose bodies could tolerate that fluctuation without severe metabolic instability would pass on their genes. The archaeological record, therefore, does more than document ancient behavior. It contextualizes the selective pressures that shaped metabolism. Constant food was not a stable backdrop across evolutionary time. It was largely absent. The evidence carved into sediment and stone reveals a long partnership between humans and variability. To continue understanding why the human body was never designed for constant food, we must now examine how demographic patterns and life expectancy influenced which metabolic traits endured through successive generations. When food arrives in waves rather than in steady lines, The body does not merely react in the moment, it begins to anticipate the pattern. Over tens of thousands of years, human metabolism adapted not just to scarcity, but to oscillation. The alternation between abundance and absence became a structural feature of life. This rhythm, sometimes described as feast and famine, was not an extreme event reserved for disaster. It was woven into ordinary existence. To understand why the human body was never designed for constant food, we must look closely at how this oscillation shaped biological expectation. In early hunter-gatherer societies, successful procurement could produce temporary abundance. A large animal provided protein and fat in quantities far beyond a single meal. Groups likely consumed significant portions quickly, both to prevent spoilage and to share within the community. After such periods of relative plenty, intake often declined. Days might pass with smaller catches or limited plant availability. This was not necessarily starvation, it was fluctuation. The body experienced cycles of higher intake followed by moderate or reduced intake. Over time, this pattern trained metabolic systems to expand and contract efficiently. Around 150,000 years ago, climatic variability intensified across parts of Africa. Paleoclimate records show alternating wet and dry phases that reshaped vegetation and animal distribution. During wetter intervals, plant resources expanded and grazing animals multiplied. During drier intervals, both contracted. Human groups living within these ecosystems would have experienced corresponding changes in food access. This environmental oscillation reinforced the feast and pause rhythm at a generational scale. Individuals whose bodies managed these swings without severe metabolic disruption were more likely to survive and reproduce. The biological mechanisms that supported this adaptability include not only fat storage and mobilization, but also shifts in resting energy expenditure. Research in modern physiology shows that during reduced intake, the body can modestly lower energy output, conserving resources without immediate collapse. This capacity likely conferred advantage in ancestral settings. It allowed individuals to remain active during lean periods while preserving vital function. The rhythm did not demand constant high intake, it demanded flexibility. Archaeological findings align with this interpretation. At several Paleolithic sites, faunal remains indicate episodic hunting success rather than evenly distributed small kills. The scale of some butchery events suggests communal consumption following significant procurement. After such events, evidence often thins. The pattern implies bursts of caloric intake interspersed with quieter phases. Even plant use shows seasonality in many regions, with certain seeds or fruits appearing only in particular layers corresponding to specific environmental windows. This structural irregularity would have reinforced metabolic cycling across generations. The concept of feast and famine should not be reduced to extremes of overindulgence and starvation. In many cases, famine meant relative scarcity rather than absolute absence. Caloric intake may have dipped, but not disappeared entirely. The important feature is variability. The body adapted to expect variation rather than constancy. Hormonal systems regulating appetite, insulin sensitivity, and energy storage evolved within this dynamic framework. They were calibrated for shifts. Around 60 to 80,000 years ago, as humans migrated into new territories, this oscillatory pattern likely persisted. Different ecosystems introduced different rhythms, but few offered uninterrupted abundance. Even in resource-rich zones, competition with other predators and climatic fluctuation introduced unpredictability. The repeated exposure to change embedded expectation into physiology. Constant food, by contrast, represents a stable plateau rarely encountered in deep time. The modern environment, with year-round access to calorie-dense options, disrupts the ancestral oscillation. The feast phase remains frequent, while the pause phase often shortens, yet the body's internal architecture still reflects its long apprenticeship to fluctuation. The metabolic systems that once thrived on cycles now operate within continuity. This mismatch does not imply inevitability of harm, but it highlights divergence between design and setting. The human organism was shaped to handle waves. It was not structured around an endless tide. As we continue exploring this rhythm, the focus will shift toward how these oscillations influence not only metabolism, but also social organization and generational survival. Because feast and pause were not merely biological experiences, They structured community life and filtered which traits endured across time. The rhythm of abundance and pause did not only shape metabolism, it shaped behavior, cooperation, and even the way communities organized themselves. In small foraging bands, a successful hunt often required coordination. Tracking, surrounding, and killing large prey demanded collective effort. When success came, the reward was shared. Sharing was not merely generosity, it was risk management. In an environment where intake fluctuated, cooperation buffered unpredictability. One individual's success today could offset another's failure tomorrow. This social distribution reinforced the feast and pause cycle at the group level. Abundance was temporary and collective. Scarcity was also collective. Together, these conditions influenced which biological traits endured. To understand why the human body was never designed for constant food, we must recognize that feast and pause were embedded not only in physiology, but also in social structure. Around 200,000 years ago, early Homo sapiens populations were small enough that cooperative survival increased the likelihood of gene transmission. Individuals whose bodies tolerated brief scarcity without rapid physical decline were better able to contribute during lean phases. They could continue moving, hunting, and participating in group life, even when intake was reduced. Over generations, resilience during pauses became part of the selective filter. The body's capacity to lower non-essential energy expenditure and to mobilize stored fat supported social continuity. A person who could remain functional during intermittent intake contributed more consistently to the group's survival. Archaeological evidence of shared butchery sites supports this interpretation. Large animal remains often show systematic distribution patterns, suggesting that meat was divided rather than consumed by a single individual. Such sharing would have spread caloric peaks across multiple bodies. When no large prey was available, reliance shifted toward gathered plants or smaller animals, often in lower quantities. The social rhythm mirrored the ecological rhythm. There were days of collective abundance and days of modest intake. Over thousands of years, this oscillation became ordinary. Its structured expectation, even reproductive patterns, were likely influenced by this variability. Periods of abundance may have supported higher fertility or improved infant survival, while lean periods imposed constraints. The interplay between energy availability and reproductive success further reinforced the value of metabolic flexibility. Individuals whose physiology adapted smoothly to oscillation were more likely to see offspring survive to maturity. Around 100,000 years ago, as humans spread into diverse climates, feast and pause rhythms varied in length and intensity, but rarely disappeared. Coastal environments might offer more frequent access to marine resources, yet storms and seasonal cycles introduced interruption. Inland environments offered large game, but also long gaps between successful hunts. Across these settings, continuous daily abundance was rare. The body did not need to adapt to uninterrupted intake, it needed to manage variation. Even today, the endocrine responses governing appetite and energy release reflect this ancestral calibration. Hunger rises and falls in patterns influenced by time since last intake, activity, and stored energy levels. The body expects change. It anticipates that intake will fluctuate. When constant food becomes available, this expectation does not vanish. The storage phase may dominate if pause intervals shrink. The social buffering that once accompanied feast and pause has transformed in modern societies into supply chains and refrigeration. Yet the biological foundation remains. The feast and pause rhythm was not a temporary stage in human history. It persisted for the overwhelming majority of our species' existence. Recognizing this continuity clarifies the central argument. The human organism was shaped within oscillation. Constant food is a recent development layered upon ancient design. As we continue, attention turns toward how the agricultural transition began to modify this rhythm without fully replacing it, because predictability entered human life gradually, rather than suddenly. The oscillation between abundance and pause did more than shape metabolism and cooperation. It shaped expectation across generations. When a pattern repeats long enough, it becomes invisible. For early humans, fluctuation was not unusual. It was ordinary. Children were born into communities where some days brought plentiful intake and other days required patience. Over time, the body's regulatory systems adapted not just to survive scarcity, but to anticipate it. This anticipation became embedded in hormonal signaling, energy allocation, and appetite control. To understand why the human body was never designed for constant food, we must see how deeply this rhythm penetrated generational experience. Across roughly 200,000 years of Homo sapiens history, before agriculture reorganized subsistence, the majority of human lifetimes unfolded within ecosystems governed by climate variability. Paleoclimate records show repeated cycles of rainfall expansion and contraction across Africa, shifts in temperature across Eurasia, and glacial oscillations that altered vegetation and animal populations. Each of these changes influenced food supply. Human groups adapted through mobility, diversified hunting strategies, and flexible gathering practices. The body, in parallel, retained the capacity to adjust internally when external supply faltered. Over many generations, those who could not tolerate intermittent intake were less likely to reproduce successfully. The filtering process of natural selection does not require dramatic famine to operate. Moderate but repeated variation is sufficient. Even small advantages in energy efficiency or storage capacity could influence survival across lean intervals. In such a context, metabolic systems favoring cyclical use of stored energy would gradually become widespread. This does not imply that prehistoric humans experienced constant deprivation. It implies that they experienced variability often enough to shape biology. Archaeological evidence of settlement patterns supports this interpretation. Many Paleolithic sites show temporary occupation followed by relocation. These patterns reflect ecological shifts rather than stable, year-round resource abundance. If food had been constant within a single territory, long-term sedentary settlement would likely have appeared earlier. Instead, mobility remained adaptive until the Neolithic transition roughly 10 to 12,000 years ago. Mobility reinforces oscillation. When groups moved in response to resource change, intake patterns shifted as well. Such recurring shifts influenced which physiological traits persisted across generations. The endurance of fat storage mechanisms, appetite cycles, and metabolic flexibility suggests that fluctuation was not a brief historical stage. It was a defining condition. Even after the development of early agriculture, variability did not vanish. Crop failure, drought, and limited storage technology continued to produce periodic scarcity. Only in very recent centuries has continuous large-scale food availability become common in parts of the world. Compared to the deep timeline of human evolution, this change is abrupt. Evolutionary processes that shaped metabolic regulation operated across tens of thousands of years. Modern abundance has existed for a fraction of that span. The body we inhabit still reflects its long apprenticeship to oscillation. Hormonal rhythms that evolved under feast and pause conditions continue to function. Energy storage pathways remain efficient. Appetite signals rise and fall in patterns calibrated for variation. These features are not relics of a distant past. They are active elements of present physiology. When food becomes constant, the ancestral rhythm does not disappear. It becomes compressed or disrupted. The design remains cyclical even if the environment becomes steady. Recognizing this generational imprint clarifies the central theme. The human body was shaped within a world of waves, not within a world of uninterrupted supply. As we move forward, attention shifts toward the agricultural transition and how the gradual stabilization of food sources began to alter, but not fully replace, this ancient rhythm. For most of human history, the internal chemistry of the body shifted in response to absence as often as it responded to intake. The endocrine system learned to interpret pauses between meals as routine rather than alarming. At the center of this regulation stands insulin, a hormone that coordinates how glucose is used and stored. Insulin does not exist in isolation. It rises when food enters the bloodstream and falls when intake pauses. In a world shaped by oscillation, this rise and fall followed a predictable ecological rhythm. To understand why the human body was never designed for constant food, we must examine how insulin and glucose regulation evolved within that fluctuating framework. When early humans consumed a successful hunt or gathered carbohydrate-rich plants, blood glucose increased. Insulin was released to guide glucose into muscle and liver cells, where it could be used immediately or stored as glycogen. Excess energy was converted into fat. This response was adaptive because abundance was temporary. The body captured what it could in preparation for interruption. After several hours, as no new food arrived, insulin levels declined. The liver began releasing stored glucose to maintain stability. Muscles shifted toward fatty acids for fuel. This alternation between insulin-dominant and insulin-low states was not rare. It was central to daily existence across tens of thousands of years. Around 100,000 years ago, as human groups expanded into new territories with varied climates, the pattern of intake remained irregular. Some days involved substantial protein and fat from large game. Other days relied on gathered plants or minimal intake. The endocrine system had to respond dynamically. Insulin sensitivity allowed tissues to absorb nutrients efficiently during abundance. Insulin decline allowed stored energy to become accessible during pause. This cycling supported survival across environmental variability. Archaeological evidence reinforces the likelihood of such metabolic fluctuation. Concentrated butchery sites indicate episodic caloric influx. Seasonal plant remains suggest periods of higher carbohydrate consumption followed by leaner months. The body adapted to expect these waves. It was not calibrated for continuous glucose elevation throughout the waking day. If constant food had characterized evolutionary history, insulin patterns might have evolved differently. Instead, what we observe in modern physiology is a hormone designed to rise and fall repeatedly. Even in contemporary settings, insulin levels decrease when intake stops for several hours. This decline is not pathological. It is functional. It allows the body to shift from storage mode to release mode. The problem is not the existence of insulin. It is the compression or elimination of the low insulin window in environments where food is available at all times. In ancestral contexts, the low insulin state occurred naturally between procurement events. In modern contexts, frequent snacking or extended eating periods can shorten that interval. The biological system itself has not fundamentally changed. It still expects oscillation. Over roughly 200,000 years, natural selection reinforced insulin regulation that accommodated fluctuation. The endocrine response was tuned to cycles of feast and pause. Continuous caloric input across the entire waking day would have been rare. Therefore, the metabolic machinery did not evolve under pressure to handle constant elevation of blood glucose without extended recovery periods. This does not imply that the body cannot tolerate modern patterns. It implies that its regulatory architecture emerged in a different context. As we continue, we will examine how this shift from oscillation to continuity alters the balance between storage and release within the body. Because insulin and glucose regulation sit at the center of the question, what happens when a cyclical system is placed in an environment of near constant supply? When the ancestral rhythm of rise and fall governed daily life, insulin was only one element within a broader network of signals. Glucagon, cortisol, growth hormone, and other regulators interacted to maintain balance across changing intake. In a fluctuating environment, these hormones operated like alternating currents. When food entered the bloodstream, insulin dominated, guiding nutrients into storage and immediate use. When hours passed without intake, glucagon signaled the liver to release stored glucose. Cortisol and growth hormone subtly adjusted energy availability, helping sustain alertness and physical capacity. This coordinated interplay reveals a system built around transition rather than permanence. To understand why the human body was never designed for constant food, we must observe how deeply this hormonal choreography depends on alternating states. Roughly 200,000 years ago, when Homo sapiens populations were small and mobile, daily life involved extended physical effort, interspersed with waiting and searching. The hormonal shifts between fed and unfed states would have supported these activities. During a successful hunt, insulin facilitated rapid storage of surplus energy. Later, during tracking or travel without immediate intake, falling insulin and rising glucagon enabled stored reserves to be accessed smoothly. These transitions were not extreme. They were moderate adjustments repeated frequently across the lifespan. Over generations, such repetition shaped endocrine sensitivity. Tissues became responsive to insulin during abundance and responsive to release signals during pause. Archaeological evidence of episodic consumption reinforces the likelihood of these repeated hormonal oscillations. Concentrated butchery sites imply short intervals of elevated caloric intake. Seasonal plant remains indicate variation in carbohydrate exposure. These environmental patterns would have produced corresponding internal fluctuations in glucose and insulin levels. The body that evolved under these conditions expected its hormonal landscape to change several times across days and weeks. Constant food, by contrast, compresses the interval between these hormonal phases. If intake continues throughout the waking day, insulin may remain elevated for longer periods than was typical in ancestral contexts. The low insulin window during which stored energy becomes accessible may shorten. Yet, the underlying design of the endocrine system still assumes oscillation. It retains the capacity to transition because that capacity was essential across deep time. Evolution does not remove such mechanisms quickly, especially when the environmental shift toward constant availability is extremely recent on a geological scale. Approximately 10 to 12,000 years ago, early agriculture began to stabilize food supply in certain regions. Even then, daily intake was structured by labor demands and limited preservation methods. Harvest cycles, crop failures, and seasonal scarcity preserved elements of fluctuation. The widespread availability of calorie-dense foods across all hours is largely a phenomenon of the last century. Compared to the 200,000 years during which hormonal regulation evolved, this interval is brief. The endocrine system still operates according to assumptions forged in oscillation. This does not imply inevitability of dysfunction, it suggests context. A system designed for alternating states may respond differently when placed in prolonged continuity. The rise and fall of insulin and its counterparts were once synchronized with ecological variation. When ecological variation diminishes, the synchronization changes. The question that follows is not whether modern food availability is good or bad. It is whether the hormonal architecture inside us was ever structured around constant stimulation. As we continue, attention turns toward how repeated compression of these oscillations may influence tissue sensitivity and metabolic balance over longer spans of time. If hormonal regulation evolved within oscillation, then the long-term balance of tissues depends not only on the presence of insulin, but on the interval during which insulin declines. In ancestral environments, that decline occurred naturally between procurement events. The body moved from storage toward release in a predictable rhythm. This rhythm shaped how cells responded to repeated stimulation. Over thousands of generations, tissues adapted to brief pulses of insulin followed by recovery periods. To understand why the human body was never designed for constant food, we must examine what this recovery interval meant at the cellular level. When insulin rises after a meal, receptors on cell surfaces respond by allowing glucose to enter. This interaction is not static. Receptors can adjust their sensitivity depending on how frequently they are stimulated. In a fluctuating environment, stimulation was intermittent. After abundance, insulin levels would fall for hours or longer, allowing receptor systems to reset. This resetting is part of normal physiology. It maintains responsiveness. In a landscape defined by feast and pause, repeated cycles of stimulation and rest would have preserved cellular balance. Around 200,000 years ago, as Homo sapiens navigated unpredictable ecosystems, this pattern unfolded countless times within individual lifespans. A hunting success led to temporary abundance and hormonal elevation. Subsequent days of reduced intake allowed levels to decline. The alternation between these states likely supported metabolic flexibility, enabling tissues to respond efficiently when nutrients were available and to rely on stored reserves when they were not. Archaeological evidence of episodic consumption supports the plausibility of such internal cycles. Concentrated animal remains and seasonal plant deposits indicate periods of elevated intake separated by intervals of scarcity. These environmental pulses would have been mirrored by endocrine pulses within the body. The cellular machinery that regulates glucose transport and energy use evolved within this context. Continuous exposure to elevated insulin across extended waking hours would have been rare. Therefore, the evolutionary pressure favored systems optimized for alternation rather than persistence. Even after the agricultural transition roughly 10 to 12,000 years ago, intake patterns retained structure. Meals were tied to labor cycles and daylight. Food preservation improved gradually but did not eliminate fluctuation. Extended fasting during seasonal shortages or cultural practices maintained intervals without intake. Only in the last century has constant access across all hours become common in certain societies. Compared to the deep timeline of metabolic evolution, this shift is abrupt. The cellular systems governing insulin sensitivity have not undergone fundamental redesign within such a brief span. They still operate according to principles shaped under oscillation. When intake becomes nearly continuous, the recovery interval shortens. The rise phase persists, but the fall phase may compress. This does not automatically produce disorder. It highlights divergence between design context and modern environment. A cyclical system placed within continuity may respond differently over time. The body that once relied on predictable hormonal descent between procurement events now navigates an environment where food can appear repeatedly without extended pause. Recognizing this divergence clarifies the central theme. Insulin regulation was shaped by waves of abundance and absence. It was not calibrated for endless elevation. As we move forward, attention turns toward the historical moment when human societies first began to stabilize food supply through agriculture, because that transition marks the beginning of a gradual shift from ecological oscillation toward greater predictability, though not yet toward true constancy. For most of human existence, food was pursued rather than planted. Mobility allowed adaptation to shifting herds, ripening plants, and changing climates. Then, roughly 10 to 12,000 years ago, in several regions of the world, a profound transition began. Humans started to cultivate, grains were sown, animals were domesticated, and settlements became more permanent. This agricultural shift did not immediately create constant food, but it altered the structure of access. To understand why the human body was never designed for constant food, we must examine how agriculture began to modify the ancient rhythm without fully replacing it. Early agricultural communities in the Fertile Crescent, parts of East Asia, Mesoamerica, and other regions depended on seasonal cycles. Crops were planted, tended, and harvested according to climate. Harvest periods produced relative abundance. Storage methods, such as early granaries and pottery, allowed some of that abundance to extend beyond immediate consumption. Yet, this predictability was limited. Harvest failure due to drought, flood, or pest could produce scarcity. Even successful harvests did not guarantee daily surplus throughout the year. Food was more stable than in purely foraging societies, but it remained vulnerable to environmental variation. The body evolving within these early farming populations still encountered oscillation. Archaeological evidence from early Neolithic sites shows the development of storage pits and communal granaries. These innovations marked a significant change. Energy could now be preserved outside the body for longer periods. However, storage capacity was finite and often communal. Loss from spoilage or contamination was common. The stabilization of supply reduced some unpredictability but did not eliminate it. Seasonal scarcity persisted, especially before the next harvest. The feast and pause rhythm became less extreme in some contexts, yet it continued to shape life. Around 8,000 years ago, as agriculture expanded across Europe and Asia, sedentary settlements grew larger. Diets became more dependent on staple grains such as wheat, barley, rice, and maize. This shift altered macronutrient patterns, increasing carbohydrate intake in many regions. Insulin responses likely became more frequent compared to earlier hunter-gatherer diets, yet meal timing remained structured by labor and daylight. Food was consumed in discrete meals rather than in continuous grazing. Even within agricultural societies, prolonged intervals without intake were common between evening and morning. The metabolic system still experienced oscillation. The transition to farming did not redesign human physiology. Evolution operates slowly. The roughly 10,000 years since widespread agriculture represent a small fraction of the 200,000 years during which metabolic regulation evolved under fluctuating foraging conditions. Some genetic adaptations to agricultural diets have occurred, such as lactose tolerance in certain populations or variations in starch digestion enzymes. However, these changes did not transform the fundamental architecture of energy storage and hormonal cycling. The body still retained its expectation of variability. Agriculture introduced greater predictability at the community level, but not uninterrupted abundance at the individual level. Crop failures, seasonal gaps, and social inequality preserved elements of scarcity. Famines are documented throughout recorded history, even in advanced agricultural societies. This historical continuity suggests that constant food, in the sense of daily and hourly access without interruption, remained rare until very recently. The agricultural revolution began a gradual shift away from the deep oscillation of foraging life. It stabilized supply in some contexts, allowing population growth and permanent settlements. Yet, it did not erase the metabolic patterns forged earlier. The human organism that moved into fields and villages carried with it an endocrine and storage system shaped by millennia of feast and pause. As we continue, attention will turn toward how industrialization and modern food systems intensified this stabilization into near continuity, creating a setting that diverges more sharply from the environment in which human metabolism first took form. As agriculture expanded and settlements became permanent, the relationship between humans and food grew more structured. Fields replaced migratory routes. Seasonal planting and harvesting began to define community calendars. Yet even within these early farming systems, access to food remained bounded by natural cycles. Grain could be stored longer than hunted meat or gathered fruit, but it was not limitless. Storage required protection from moisture, pests, and decay. Losses were common. A failed harvest could ripple through an entire village. To understand why the human body was never designed for constant food, it is essential to see that early agriculture introduced stability, but not continuity. Around 8 to 10,000 years ago, in regions such as the Fertile Crescent, early farmers relied heavily on annual crops. Harvest time marked a period of relative abundance. Following harvest, granaries filled and caloric security improved temporarily. Yet as months passed, stored grain diminished. Consumption was paced carefully to bridge the interval until the next harvest. This pacing preserved a rhythm. There were still periods of relative plenty and periods of caution. The body living in these early agricultural communities continued to experience fluctuation, though perhaps less dramatic than in pure foraging contexts. Archaeological excavations of early Neolithic settlements reveal storage pits, communal silos, and grinding tools for grain processing. These artifacts signal a shift toward planning and preservation. However, they also reveal vulnerability. Grain remains often show signs of spoilage or infestation. Entire storage caches sometimes appear abandoned, likely due to contamination. This fragility meant that agricultural predictability was partial. It reduced uncertainty but did not eliminate it. Famine and crop failure appear repeatedly in historical records as societies grew more complex. These recurrent shortages demonstrate that constancy was never guaranteed. Around 5,000 to 6,000 years ago, as agricultural societies expanded across Europe, Asia, and parts of Africa, diet composition shifted further toward carbohydrates. Regular bread or porridge meals became more common. Insulin responses may have occurred more predictably within daily cycles, yet meal frequency remained structured by labor patterns. Farming required long hours in fields, often with limited opportunities for continuous snacking. Meals were taken at set times, often 2 or 3 per day, separated by intervals of work and fasting. Nighttime always imposed a prolonged pause in intake. Even with agriculture, the metabolic system retained space for oscillation. Evolutionary change in physiology during this relatively short agricultural period was modest compared to the deep pre-agricultural timeline. Some genetic adaptations appeared, such as increased amylase gene copies in populations consuming starch-rich diets. These changes improved digestion efficiency but did not redesign the fundamental structure of hormonal cycling or fat storage. The body still expected periods when insulin declined and stored energy was accessed. The agricultural revolution stabilized supply enough to support population growth and social complexity. It did not create a world of constant food in the modern sense of round-the-clock access to calorie-dense options. That transformation required industrialization, transportation networks, and food processing technologies that emerged much later. For thousands of years after the first fields were planted, human metabolism still operated within cycles shaped by daylight, labor, and seasonal variation. The ancient rhythm was softened but not erased. Recognizing this gradual shift clarifies the argument. Agriculture marked a turning point in food predictability, yet it did not fundamentally alter the evolutionary design of the human body. The next phase of history would compress these cycles more dramatically, bringing humanity closer to uninterrupted access than ever before. As agricultural systems matured, Villages grew into towns and towns into early cities. Trade networks expanded, linking regions with different harvest cycles. Grain from one valley could be exchanged for goods from another. In some areas, irrigation reduced dependence on rainfall alone. These developments gradually buffered communities against local crop failure. Yet even as food distribution became more organized, constancy at the level of individual intake remained limited. To understand why the human body was never designed for constant food, it is important to see that predictability increased slowly and unevenly across history, and rarely reached uninterrupted continuity. Around 4,000 to 5,000 years ago, early urban centers in Mesopotamia, Egypt, and the Indus Valley relied on centralized storage and redistribution. Written records from these societies document rations allocated to workers and officials. This administrative structure suggests greater control over supply than in earlier farming villages. However, these systems were vulnerable to environmental stress and political instability. Drought, flooding, conflict, or trade disruption could rapidly destabilize food access. Historical records from ancient civilizations frequently mention famine. Even organized states could not guarantee daily abundance for all members of society. Social hierarchy often determined who ate reliably and who did not. The metabolic design of the average person remained exposed to variability. Archaeological evidence from urban sites reveals both storage facilities and signs of malnutrition in skeletal remains. These findings indicate that while food preservation improved, access was not universally stable. Seasonal rhythms continued to influence availability. Harvest time brought greater abundance. Late winter or early spring, before new crops matured, often produced scarcity. This pattern persisted across centuries. The feast and pause cycle became less tied to hunting success, and more tied to agricultural timing, yet oscillation endured. Even in societies with advanced granaries, food preservation was imperfect. Rodents, insects, mold, and spoilage reduced supplies. Surplus could not always be extended indefinitely. Furthermore, daily routines were structured around work demands. Meals were consumed at specific times rather than continuously. Nighttime fasting remained unavoidable in the absence of artificial lighting and refrigeration. The endocrine system of individuals living in these societies still experienced daily declines in insulin between meals and overnight. The body's expectation of oscillation remained aligned with lived reality. Approximately 200,000 years of evolutionary conditioning had shaped metabolism long before the rise of cities. The several thousand years of early civilization were not sufficient to erase that conditioning. Although genetic adaptations to diet continued in limited ways, the fundamental architecture of energy storage and hormonal cycling persisted. Even as agriculture and urbanization increased stability at the population level, individual physiology retained its ancestral calibration. Only in the last few centuries, with the advent of industrial agriculture, mechanized transport, and eventually globalized food systems, did the possibility of near-constant access begin to materialize. Compared to the immense span of pre-agricultural and early agricultural history, this modern development is brief. The body that enters supermarkets and refrigerators today still carries the imprint of oscillation. It was shaped in savannas, fields, and early cities where supply rose and fell with season, labor, and climate. Recognizing this continuity across epochs clarifies the central theme. Agriculture and early civilization softened unpredictability, but did not eliminate it. The human organism evolved within cycles that persisted far longer than the recent experience of abundance. As we move forward, attention shifts toward the industrial transformation that intensified food availability into a level of constancy unprecedented in human history. The transformation from seasonal stability to near permanence did not occur with the first farms or even the first cities. It accelerated much later. During the industrial era, roughly 2 to 3 centuries ago, mechanization began reshaping agriculture. Steam power, improved tools, and later fossil fuel-driven machinery increased yield and reduced dependence on manual labor. Transportation networks expanded through railways and steamships, allowing grain, sugar, and other staples to move across continents. For the first time in human history, Local crop failure did not always mean immediate scarcity if trade routes remained intact. This development marked a profound shift. Yet even here, the body that encountered this new environment remained largely the same body shaped across 200,000 years of oscillation. To understand why the human body was never designed for constant food, we must see how abruptly industrialization intensified availability compared to evolutionary time. In pre-industrial societies, even with agriculture, food was tied closely to daylight and labor cycles. Meals were structured around work and rest. Night imposed an unavoidable pause. Refrigeration did not exist. Preservation methods such as salting, drying, and fermenting extended supply, but did not allow effortless, continuous access. The Industrial Revolution changed these constraints. Mechanized milling increased flour production. Refined sugar became more widespread. Urban populations grew, and markets operated daily. As the 19th century progressed, railways transported food rapidly from rural areas to cities. The spatial limits that once constrained availability weakened. Around the late 19th and early 20th centuries, refrigeration technology emerged. Cold storage extended the shelf life of perishable goods. Meat and dairy could be preserved and transported over long distances. Seasonal limitations diminished. A person in an industrializing city could obtain foods previously restricted to particular times of year. This change compressed the ancient feast and pause cycle even further. Instead of waiting for harvest or migration, individuals encountered a steady flow of calories. The metabolic system, however, had not been redesigned to anticipate uninterrupted intake. Evolution does not operate on the timescale of centuries. The hormonal and storage mechanisms shaped in fluctuating environments remained intact. Industrialization did not remove oscillation entirely. Many working-class families in early industrial cities still face poverty and food insecurity. Yet for increasing segments of society, especially in wealthier regions, food became more consistently accessible. The rhythm of intake began to detach from ecological cycles and attach to economic ones. Work schedules, factory shifts, and urban markets structured meals. Instead of eating primarily when food was procured, people began eating according to clock time. Breakfast, lunch, and dinner became standardized patterns in many societies. This temporal restructuring altered daily hormonal cycles. Insulin still rose and fell, but now in relation to fixed hours rather than unpredictable procurement. Archaeological and historical records show that caloric intake increased in many industrializing regions. Refined carbohydrates and processed foods became more common. These foods often produce rapid increases in blood glucose. The frequency and intensity of insulin stimulation may have risen compared to earlier agricultural patterns. Yet overnight fasting still occurred. Meals were discrete. Constant grazing throughout the waking day was not yet universal. The full compression of oscillation required additional developments in food processing, marketing, and cultural norms. Industrialization laid the foundation by weakening ecological constraints and strengthening supply chains. It moved human societies closer to constancy than ever before. The body entering this era carried ancient assumptions about variability. The environment was beginning to diverge more sharply from those assumptions. As we continue, attention turns toward how the 20th century and the rise of highly processed, continuously available foods intensified this divergence, creating conditions far removed from the oscillatory world in which human metabolism first took shape. As industrial systems matured in the 20th century, the separation between food and season widened further. Mechanized agriculture increased yield beyond what earlier societies could sustain. Synthetic fertilizers, improved crop varieties, and large-scale irrigation amplified production. Refrigerated transport and global shipping networks made it possible to consume fruits, grains, and animal products far from their place of origin and far from their natural season. This expansion did not merely increase supply, it changed expectation. For the first time in human history, significant portions of the population in certain regions could assume that food would be available at almost any hour. To understand why the human body was never designed for constant food, we must consider how radically this shift diverges from the deep patterns that shaped metabolism. In earlier agricultural societies, even with storage, food required preparation and planning. Meals were structured events. Snacking between them was limited by practicality. The 20th century introduced packaged and processed foods designed for convenience. Shelf-stable products could be stored in homes for extended periods. Supermarkets replaced local markets, offering wide variety under one roof. Vending machines and later convenience stores extended access beyond traditional meal times. The interval between procurement and consumption shrank dramatically. Instead of walking, hunting, harvesting, or preparing extensively, individuals could obtain calories with minimal effort. This compression of effort altered daily rhythms. Around the mid-20th century, marketing practices and work schedules reinforced the idea of frequent eating. Advertisements promoted snacks between meals. Work environments often included brief breaks accompanied by quick consumption. Over time, the cultural norm in some societies shifted toward eating multiple times throughout the day. The ancient oscillation of feast and pause began to flatten. Insulin still rose in response to intake, but the duration of low insulin intervals shortened when meals and snacks were spaced closely together. The endocrine system itself did not change fundamentally. It continued to operate as it had for thousands of generations. What changed was the frequency of stimulation. Compared to the roughly 200,000 years during which humans navigated unpredictable environments, the widespread availability of processed, ready-to-eat food is extraordinarily recent. Evolutionary biology teaches that traits shaped across deep time do not recalibrate instantly in response to rapid environmental change. The metabolic architecture designed for waves now encounters near continuity in some settings. This does not mean that all individuals experience identical outcomes. Socioeconomic differences, cultural practices, and individual behaviors create variation. Yet at a structural level, the environment in many industrialized regions now permits constant access in a way that would have been unimaginable for most of human history. Archaeological layers from earlier periods reveal pulses of consumption tied to ecological cycles. Modern supermarkets represent a different archaeological layer, one characterized by storage, refrigeration, and global distribution. The shift from ecological limitation to economic and logistical abundance marks a turning point in the relationship between humans and food. The body that evolved under oscillation now inhabits a world where oscillation is optional rather than inevitable. The next step in this historical progression intensified availability even further, as food processing technologies refined texture, flavor, and shelf life, encouraging more frequent consumption. To understand the full divergence between ancient design and modern context, we must examine how these technologies reshaped not only supply, but also desire and habitual intake patterns. By the late 20th century, the transformation was no longer only about supply. It was about environment. Food ceased to be something encountered at specific times and places. It became embedded in daily surroundings. Refrigerators stood within arm's reach in private homes. Processed snacks required no preparation. Restaurants extended hours into the night. In many urban centers, food could be purchased at any hour. This environmental saturation represents a departure from the conditions under which human metabolism evolved. To understand why the human body was never designed for constant food, we must consider how continuous availability reshapes daily biological rhythms that once depended on unavoidable gaps. For most of the 200,000 years of Homo sapiens history, darkness imposed a pause. Without artificial light and preservation, nighttime eating was rare. The endocrine system experienced extended low insulin intervals between the final meal of the day and the next opportunity for procurement. These overnight windows allowed stored energy to be accessed, and metabolic processes to shift naturally. Even during early agricultural and industrial periods, this nightly pause remained intact. It was only with electrification, refrigeration, and round-the-clock commerce that the possibility of eliminating this pause became common in certain societies. The compression of oscillation is therefore both spatial and temporal. Spatially, food is now present in nearly every environment: workplaces, transportation hubs, entertainment venues. Temporally, access extends across the entire waking period, and sometimes beyond. This does not force constant eating, but it removes structural barriers that once enforced intervals. The body's hormonal architecture, however, still reflects ancient cycles. Insulin, glucagon, and related signals continue to operate as though pauses are expected. When intake occurs frequently, the low insulin state may shorten, yet the underlying design has not been fundamentally altered. Around 100 years is negligible in evolutionary time. The genetic and physiological traits shaped across millennia remain largely intact. The modern environment, therefore, represents a divergence rather than a redesign. Archaeology provides a useful contrast. Earlier layers show episodic evidence of consumption linked to ecological opportunity. Modern food systems leave a different kind of trace. Packaging waste, storage infrastructure, and global transport networks. These traces document a world in which constancy is technologically enabled. The human organism, however, did not evolve under such enabling conditions. It evolved under constraint. Constraint created rhythm. Rhythm shaped metabolism. When constraint diminishes, rhythm can become optional. The question is not whether modern abundance is inherently harmful. It is whether the body was structured to assume it. The design of energy storage, hormonal cycling, and appetite signaling suggests expectation of fluctuation rather than permanence. Constant food, in the sense of uninterrupted accessibility and frequent intake, is a recent overlay upon an ancient system. The divergence between environment and design has widened most dramatically in the last century. As we move forward, attention shifts toward how this divergence interacts with population structure and longevity, because humans now live longer than most of their ancestors, carrying metabolic systems shaped in oscillation across decades of sustained abundance. As food became continuously available in many modern societies, another transformation unfolded quietly. Eating shifted from being an event tied to procurement or structured meals into an activity woven throughout the day. The rise of snacking culture did not happen all at once, but gradually, through changes in work patterns, marketing, and convenience. To understand why the human body was never designed for constant food, we must examine how the normalization of frequent intake compresses the ancient rhythm of oscillation into near continuity. For most of human history, meals were bounded by effort. Hunting, gathering, planting, harvesting, or preparing food imposed natural intervals. Even in agricultural and early industrial societies, meals were distinct episodes separated by hours of labor. The endocrine system rose and fell in response to these episodes. Insulin increased during intake and declined between meals, allowing stored energy to be accessed. These low-intake windows were not deliberate health practices. They were built into the structure of daily life. In contrast, modern environments often encourage small, frequent eating occasions. Processed foods designed for portability and shelf stability make consumption possible in moments that once would have been empty. Marketing campaigns promote snacks as necessary boosts for energy or focus. Workplace routines include brief breaks accompanied by packaged foods. Children grow up with the expectation of multiple eating occasions beyond traditional meals. The cumulative effect is a shortening of the interval between insulin rises. The body's hormonal architecture remains cyclical, but the cycle tightens. Around 200,000 years ago, such a pattern would have been rare. Food access was constrained by ecology. Even after agriculture, intake frequency was limited by availability and preparation time. The normalization of eating across nearly the entire waking day represents a distinct departure from ancestral conditions. This does not mean that occasional snacks are incompatible with human biology. It suggests that persistent stimulation throughout the day differs from the oscillation that shaped metabolic design. Archaeological evidence reinforces the contrast. Earlier layers reveal concentrated consumption events and clear pauses marked by mobility or environmental change. There is little sign of constant, low-level intake distributed evenly across time. The body that evolved under those conditions expects phases of storage and phases of release. When eating becomes nearly continuous, the release phase may diminish in duration. Insulin remains elevated more often, and the low insulin window narrows. Evolutionary change does not track cultural shifts at the pace of decades. The metabolic systems forged across millennia still operate within their inherited framework. Snacking culture illustrates how environment can reshape behavior without immediately reshaping biology. The availability of calorie-dense, rapidly digestible foods intensifies this effect. Refined carbohydrates and sugars produce quick elevations in blood glucose, prompting insulin release. When such stimuli occur repeatedly across the day, the oscillatory pattern becomes compressed. The body was not originally structured around constant stimulation. It was structured around cycles of effort, intake, pause, and mobilization. Recognizing this helps clarify the central argument. The rise of frequent, convenience-based eating is not simply a cultural preference. It represents an environmental condition that diverges from the oscillatory landscape in which human metabolism first took form. As we continue, attention turns toward how this persistent compression of oscillation may interact with long-term tissue sensitivity and metabolic balance across decades of extended lifespan. As frequent eating becomes normal, the meaning of hunger quietly changes, not because the body has been redesigned, but because the environment now answers the signal almost instantly. In the deep past, hunger rose inside a world that did not respond on demand. A person could feel the first pull of appetite and still have no immediate option except to keep walking, keep scanning the ground, keep following tracks, or keep working with others. That delay mattered. It gave the body time to complete a natural sequence. Insulin gradually falling, stored glycogen being released, fat beginning to contribute more, and attention narrowing towards seeking resources. In that older setting, hunger was not only discomfort, it was a guide that unfolded over time. Today, in many modern lives, the same signal is often treated as a problem to erase within minutes. A snack in a pocket, a vending machine in a hallway, a drive-through on the road, a refrigerator a few steps away, all shorten the distance between cue and consumption. This does not change the hormone systems themselves, but it changes how often they can cycle through their full range. The body begins a transition toward using stored energy, and then the transition is interrupted. Over and over, the rhythm that once moved from intake to pause to mobilization becomes a rhythm of repeated intake with only shallow pauses. This is important for the central question, because a design shaped for oscillation depends on having space for oscillation to occur. 200,000 years ago, the interval between hunger and food was often long enough to be meaningful. It might be an hour while searching for tubers, several hours while tracking an animal, or longer across unsuccessful attempts. During those intervals, the body did not simply wait helplessly. It maintained blood glucose through liver release, shifted muscles toward different fuels, and kept the brain supplied. That ability was selected for because it supported survival in real conditions. Hunger did not always lead to immediate eating, and the body did not require immediate eating to function. Archaeological patterns fit this logic. Many sites show concentrated evidence of butchery and cooking, rather than a continuous scatter that would suggest constant nibbling. Hearth layers, bone deposits, and tool clusters imply episodes of consumption separated by time and movement. Even in plant use, seasonal traces suggest periods of higher intake and periods of lower intake, not a constant trickle. These material patterns do not tell us how often a person felt hunger, but they strongly suggest that hunger frequently had time to persist, to rise and fall, and to be matched by internal mobilization before being satisfied. Over many generations, this pairing of signal and delay shaped expectations in physiology. The body's hunger signals are not built on the assumption that every cue will be answered immediately. They are built to tolerate waiting. That is why the sensation tends to come in waves rather than a straight line of panic. In modern settings, repeated instant responses can change habits and perception. If the first small cue is always answered, the person may rarely experience the later phase of the wave where the signal can soften as the body settles into a different fuel balance. This does not mean hunger should be prolonged as a rule, and it does not claim that everyone responds the same way. It simply observes a mismatch. The signal evolved with delay as a normal feature, while the environment now allows delay to vanish. Even after agriculture began, meals were still structured by work, daylight, and preparation time. Food might be more predictable across seasons than in foraging life, but it was not typically eaten in constant small increments all day. Night still created a long pause. Early industrial life for many people still centered around discrete meals rather than continual grazing. The widespread presence of portable processed foods made grazing easier, and then cultural norms grew around it. In that shift, hunger becomes less like a guide through a variable landscape and more like a button that triggers immediate consumption. If the human body was designed for cycles, then the repeated removal of the pause portion changes the daily shape of those cycles. It does not erase ancient design, but it can keep parts of it from being expressed. This is one of the clearest ways modern constant access differs from the conditions that shaped us, because it changes not just what we eat, but how often the body is allowed to move from one internal state to another. If frequent eating compresses the ancient rhythm of hunger and release, the question becomes larger than a single day. It becomes generational. For most of human history, individuals did not live long enough to experience decades of sustained abundance layered on top of a metabolism shaped for oscillation. Average life expectancy in prehistoric populations was low, largely due to infant mortality, infection, and injury. Those who survived childhood could live into middle age and sometimes beyond, but few experienced 70 or 80 years of continuous food access. The modern world has changed this dramatically. To understand why the human body was never designed for constant food, we must consider how extended lifespan interacts with compressed metabolic cycles. Around 200,000 years ago, the selective pressures acting on metabolism were concentrated in early and middle life, the years most relevant to reproduction and child-rearing. The body's ability to endure fluctuating intake during these phases increased the likelihood of passing on genes. Long-term exposure to uninterrupted abundance across many decades was not part of the evolutionary testing ground. Even after agriculture and early urbanization, recurrent scarcity, labor demands, and seasonal gaps preserved elements of fluctuation throughout much of life. Only in the last century have significant portions of some populations combined two factors: long lifespan and nearly continuous access to calorie-dense food. This combination is historically new. The endocrine and storage systems operating within modern adults are still those shaped by deep time. Insulin rises and falls according to intake. Fat is stored efficiently during surplus. Appetite signals respond to cues. Yet when cycles are repeatedly compressed over decades rather than years, the cumulative pattern differs from ancestral conditions. The body that evolved to handle waves across a shorter expected lifespan now experiences prolonged exposure to continuity. Archaeological and demographic evidence helps frame this contrast. Skeletal remains from prehistoric populations often show signs of physical stress and injury, but relatively low evidence of chronic metabolic diseases associated with long-term abundance. This does not mean such conditions were absent, only that they were less visible in populations that rarely reached advanced age under stable caloric excess. In contrast, modern societies with industrialized food systems and medical advances allow individuals to live far beyond reproductive years while maintaining high and frequent intake. The metabolic architecture has not been rebuilt to match this extended exposure. It still reflects assumptions forged under oscillation. This divergence does not assign blame to modern life, nor does it romanticize the past. It highlights a structural difference. The body's regulatory systems were filtered through selection pressures operating under variable supply and shorter average lifespans. The current environment introduces long-term constancy layered onto extended longevity. When daily cycles of intake are flattened and repeated across decades, the internal balance between storage and release can gradually shift. The design remains cyclical, but the expression of that cycle becomes shallow. Over years, shallow cycles repeated thousands of times may differ from deeper oscillations repeated under ancestral conditions. Recognizing this helps clarify the central theme. Constant food is not merely a matter of convenience. It represents a sustained environmental context, unlike the one that shaped human metabolism across deep time. The body we inhabit is capable of remarkable adaptation, yet it carries within it the imprint of a world defined by waves rather than permanence. As we move forward, attention turns toward what this divergence means at the cellular and tissue level over extended periods of life, because the consequences of compressed oscillation may accumulate slowly rather than appearing immediately. When lifespan extends far beyond the years in which natural selection was most active, the body begins to reveal patterns that were once less visible. For most of the 200,000 years of Homo sapiens history, survival pressures were concentrated in early life. Infant mortality was high. Infection, injury, and environmental hardship limited how many individuals reached advanced age. Those who survived childhood and reproduced passed on their genes. The metabolic systems that evolved under these pressures were optimized primarily for survival through fluctuating conditions during youth and middle age. They were not tested across decades of uninterrupted abundance combined with long post-reproductive lifespan. To understand why the human body was never designed for constant food, we must examine how population structure across deep time shaped metabolic priorities. In small foraging groups, demographic structure was uneven. Many children did not reach adulthood. Average life expectancy at birth was low, even though some individuals lived into their 60s or beyond. The majority of reproductive success occurred relatively early in life. Under such conditions, traits that supported short to medium-term resilience during ecological fluctuation were strongly selected. Efficient fat storage during abundance and effective mobilization during scarcity increased the probability of surviving to reproduce. Long-term consequences of sustained caloric excess in old age would have exerted little selective pressure because few individuals experienced that scenario consistently. Archaeological and skeletal evidence supports this demographic picture. Remains from Paleolithic contexts show signs of trauma and infectious disease, but relatively limited evidence of chronic degenerative conditions associated with prolonged metabolic surplus. This does not mean such conditions were absent. It suggests that environmental variability and shorter average lifespans limited sustained exposure. Even after agriculture began roughly 10 to 12,000 years ago, life expectancy remained constrained by disease, famine, and labor hardship. Cycles of scarcity persisted. The body's metabolic architecture continued to operate within oscillation across most of an individual's life. Only in the last 2 centuries, particularly in the last 100 years, have significant portions of the global population experienced both extended longevity and stable caloric access. Medical advances reduced mortality from infection and injury. Agricultural and industrial systems increased food availability. The demographic profile shifted toward older age groups. For the first time, large numbers of people could live into their 70s and 80s while maintaining high and frequent intake. This demographic transformation is historically unprecedented. The body, however, still carries regulatory systems shaped when reproductive success in fluctuating environments was the primary filter. Natural selection does not optimize organisms for conditions that arise suddenly and recently. It preserves traits that improve survival and reproduction in past contexts. If constant food across decades of extended life had characterized most of human evolution, different metabolic priorities might have emerged. Instead, the architecture we possess reflects assumptions of variability and finite lifespan under ecological constraint. When abundance and longevity combine, the long-term balance between storage and release can shift gradually. Tissues that were once exposed to cycles of oscillation for shorter spans now experience compressed cycles repeated across many decades. This does not imply inevitability of dysfunction, nor does it reduce modern life to a simple mismatch. It clarifies context. The human organism was shaped within a demographic structure defined by fluctuation and limited longevity. Constant food layered onto extended lifespan represents a new environment, not the one in which metabolism was forged. As we continue, attention turns toward how this demographic shift interacts with intergenerational patterns and inherited tendencies, because population structure influences not only individuals, but also how metabolic traits are expressed across families and communities. When a population shifts toward longer life expectancy, the timing of metabolic exposure changes as well. For most of human evolutionary history, the years that mattered most for gene transmission were early adulthood and middle age. After reproduction, selective pressure weakened. Traits that supported survival during childbearing years were preserved, even if they carried consequences later in life. This principle helps clarify why the human body was never designed for constant food across many decades. The metabolic systems that favored efficient energy storage and flexible fuel switching were highly advantageous in fluctuating environments. They increased the likelihood of surviving lean periods and raising offspring. Whether those same systems might respond differently after 40, 50, or 60 years of sustained abundance was largely irrelevant to selection in prehistoric contexts, because few individuals experienced that combination for long. Around 200,000 years ago, in small and mobile groups, generational turnover was rapid. Children who survived to adulthood began contributing to the next generation while their parents were still relatively young. Extended multi-generational overlap existed, but the proportion of very old individuals was small. Energy allocation within the body prioritized growth, reproduction, and immediate survival. Fat storage during abundance protected against famine and supported pregnancy and lactation. In such a demographic landscape, selection reinforced metabolic thrift rather than restraint. The ability to store quickly and mobilize effectively was adaptive. Long-term consequences of storing too efficiently in an environment of constant food were not a central concern because constant food did not exist. Even after agriculture and early urbanization, mortality from infectious disease, malnutrition, and labor strain kept average lifespan constrained. Historical records from pre-modern societies repeatedly document cycles of famine and epidemic. Although food supply was more predictable than in foraging life, it was not stable across decades for most individuals. The body still encountered oscillation, and demographic pressure still focused on early life resilience. Only in the last 1 to 2 centuries has a significant shift occurred. Public health improvements, sanitation, vaccines, and antibiotics dramatically reduced early mortality in many regions. At the same time, industrial agriculture and global distribution stabilized calorie availability. The result is a demographic profile with large proportions of older adults living in environments of steady intake. This combination creates a scenario that was rare across evolutionary time, a body shaped for thrift, operating under abundance for 70 or 80 years. Intergenerational patterns also begin to matter in new ways. Traits that once offered survival advantage during scarcity may now interact with continuous intake across multiple decades within families. However, genetic adaptation to such a rapid environmental shift is slow. The timescale of a few generations is brief compared to the tens of thousands of years during which metabolic regulation was forged. The fundamental architecture of insulin signaling, fat storage, and hunger response remains rooted in oscillation. Demographic transformation does not automatically rewrite physiology. It alters the context in which that physiology operates. Recognizing this helps clarify the argument. The human body was not selected under conditions of lifelong abundance combined with extended longevity. It was selected under conditions of fluctuating supply and shorter average lifespans. The modern demographic landscape magnifies the divergence between ancient design and contemporary context. As we continue, attention turns toward how this long-term exposure to compressed cycles influences tissues at a cellular level across decades, because the consequences of constancy may unfold slowly over time rather than appearing immediately. When demographic patterns change, biology does not instantly follow. The extension of human lifespan over the last 2 centuries has created a new context layered onto ancient physiology. For most of our evolutionary past, metabolic systems were filtered by pressures acting during youth and early adulthood. Survival to reproductive age, successful childbearing, and protection against famine were the primary tests. Traits that enhanced these outcomes persisted, even if they carried long-term costs that might appear later in life. To understand why the human body was never designed for constant food, we must see how this selective focus shaped priorities within metabolism. Around 200,000 years ago, in fluctuating African ecosystems, the body's capacity to store energy efficiently during abundance was directly tied to survival during scarcity. Individuals who could accumulate reserves and then mobilize them effectively during lean periods were more likely to reach reproductive age and raise offspring. This advantage operated repeatedly across generations. There was little selective pressure to reduce storage efficiency in anticipation of decades of uninterrupted abundance, because uninterrupted abundance was not part of the environment. Even after agriculture began, cycles of drought, flood, crop failure, and conflict preserved variability. Famine appears frequently in written records from ancient and medieval societies. Under these conditions, metabolic thrift remained adaptive. The demographic shift of the modern era introduced a scenario rarely encountered before: extended life under relatively stable caloric supply. Many individuals now live for decades beyond the typical reproductive window. During these later decades, the same storage and hormonal systems continue to function. They were not redesigned to accommodate continuous intake across 70 or 80 years. Evolutionary theory suggests that traits with effects primarily after reproduction are less strongly shaped by natural selection. If constant food across long lifespan had been common in prehistoric populations, metabolic regulation might have evolved differently. Instead, the design we observe reflects ancient pressures emphasizing flexibility during fluctuating supply in earlier life stages. Archaeological and skeletal evidence from earlier eras reinforces this perspective. While some individuals did reach advanced age, they represented a smaller fraction of the population. Many did not experience prolonged exposure to steady abundance. The combination of extended longevity and near-constant access to calorie-dense food is therefore historically new. The metabolic architecture forged across millennia now operates in an environment it did not anticipate. This does not mean the body is flawed. It means its design assumptions differ from modern conditions. When cycles of intake and pause become shallow, yet persist across decades, the cumulative effect differs from ancestral patterns, where deeper oscillations occurred within shorter lifespans. Demography amplifies environment. A cyclical system exposed to continuity for many years may behave differently than one exposed to waves across fewer years. Recognizing this helps frame the central theme without exaggeration. The human body was selected for resilience in fluctuating contexts, not for optimization under lifelong abundance. The recent demographic transformation does not invalidate ancient design, but it highlights divergence between the conditions that shaped metabolism and the conditions in which it now operates. As we move forward, attention shifts toward what happens within cells and tissues when oscillation becomes compressed repeatedly across long spans of life, because the consequences of constancy may unfold quietly and gradually over time. When oscillation shortens and intake becomes frequent across long spans of life, the effects may not appear suddenly. They unfold at the level of cells and tissues, slowly, through repeated stimulation. The human body was built to move between states. Storage and release, growth and repair, activation and rest were meant to alternate. To understand why the human body was never designed for constant food, we must look beneath daily habits and examine what happens inside tissues when the pause phase grows shallow but persists for decades. At the cellular level, energy is not only fuel, it is information. Nutrient availability signals cells to grow, divide, and synthesize proteins. Hormonal cues such as insulin activate pathways that promote storage and construction. In ancestral environments shaped by fluctuating supply, these growth signals were balanced by intervals in which nutrient levels declined. During those intervals, cells shifted toward maintenance processes. Damaged components could be broken down and recycled, energy use recalibrated. This alternation between growth and maintenance was not a conscious strategy. It was a biological rhythm aligned with feast and pause. Roughly 200,000 years ago, when early Homo sapiens moved across uncertain landscapes, these cycles would have occurred naturally. After successful intake, growth pathways activated. During subsequent hours or days of reduced intake, maintenance processes became more prominent. The balance between these states likely supported long-term resilience within the limits of prehistoric lifespan. The body expected variation. It did not expect continuous stimulation of growth signals without sufficient intervals of metabolic quiet. Even after agriculture began, daily structure preserved oscillation. Meals were separated by labor and sleep. Seasonal variation still influenced supply. Although predictability increased in some regions, the fundamental alternation between fed and unfed states remained. Only with modern constant availability has the potential arisen for growth-related signaling to remain elevated more frequently across waking hours. This does not imply that cells cease maintenance entirely. It suggests that the depth and duration of maintenance intervals may shorten, the ancient rhythm becomes compressed. Archaeological records do not show us cellular processes, but they show the context in which those processes evolved. Episodic consumption, mobility, and environmental fluctuation provided natural intervals in which nutrient signaling subsided. The metabolic pathways shaped in that context retain their cyclical logic. When intake occurs repeatedly throughout the day, insulin and related signals may activate growth pathways more often. Over years and decades, the cumulative pattern differs from one in which long pauses were routine. The body was designed to handle variation. It was not calibrated for permanent abundance at the cellular signaling level. This divergence does not guarantee a single outcome for every individual. Genetic variation, physical activity, sleep patterns, and cultural practices influence expression. However, the structural difference remains clear. A system shaped under oscillation now operates in an environment that can reduce oscillation. The question is not whether modern life is inherently harmful, it is whether the biological architecture inside us assumed cycles that are now less pronounced. As we continue, we will examine more closely how specific cellular maintenance processes interact with feeding intervals, because understanding that interaction brings us closer to the core of the original question. What happens when a body built for rhythm lives in constancy? Within each cell, balance depends on timing. Nutrients entering the bloodstream do more than provide energy. They activate signaling networks that influence how cells grow, divide, repair, and recycle their internal components. In environments shaped by fluctuation, these networks operated in alternating phases. After intake, pathways associated with growth and storage became more active. When intake paused, other pathways associated with maintenance and internal cleanup increased in relative prominence. This alternation was not designed consciously. It emerged because ecological conditions repeatedly imposed intervals between food availability. To understand why the human body was never designed for constant food, we must look closely at how these intracellular rhythms depend on the presence of genuine pause. One important aspect of cellular maintenance involves the recycling of damaged proteins and organelles. Cells continuously generate waste as part of normal metabolism. Under conditions of reduced nutrient signaling, processes that break down and reuse internal components become more active. This recycling supports long-term stability. In fluctuating ancestral environments, such periods of reduced signaling would have occurred naturally between procurement events and during overnight fasting. Roughly 200,000 years ago, when human groups moved through unpredictable landscapes, these low-intake windows were unavoidable. They created opportunities for maintenance processes to unfold without persistent stimulation from incoming nutrients. The endocrine system, through lower insulin and altered energy signaling, helped facilitate these shifts. Even during early agricultural periods, daily life preserved intervals without intake. Meals were tied to labor and daylight, and nighttime always imposed an extended break. Seasonal scarcity reinforced additional pauses. Although overall predictability increased compared to foraging life, constancy remained rare. The metabolic architecture retained its expectation of alternating states. Only in recent centuries has the possibility arisen for nutrient signaling to remain elevated across much of the waking day. Frequent snacking and extended eating windows can reduce the duration of low-signal intervals. This does not eliminate maintenance processes entirely, but it can compress their natural timing. The body's design does not assume that nutrients will flow continuously. It assumes variation. Archaeological patterns indirectly support this view by documenting episodic intake and mobility across vast spans of time. The physical remnants of prehistoric life show concentrated consumption events separated by movement and environmental shifts. Such patterns imply regular metabolic oscillation. The cellular systems that evolved within this context were shaped by repetition. Growth phases were followed by quieter intervals. Over generations, this pairing became embedded in physiology. When constant food becomes possible, the growth-promoting signals may recur more frequently without equivalent depth of pause. Over years and decades, this altered timing can influence how tissues allocate energy between building and maintenance. The design itself remains cyclical. The environment becomes less so. Recognizing this does not require moral judgment about modern eating patterns. It requires historical perspective. The human organism evolved under constraints that structured time as well as space. Constraint produced rhythm. Rhythm shaped cellular processes. When constraint diminishes, rhythm can flatten. As we move toward the final stages of this exploration, attention will turn to how these compressed cycles interact with aging across extended lifespan, because the long-term effects of constant availability may reveal themselves most clearly over time rather than in immediate experience. When we place cellular timing inside the full span of a human life, a quiet tension becomes easier to see. The body is not built to remain in one metabolic mood forever. It is built to move between states, like a landscape that repeatedly shifts from wet to dry, from growth to repair, from storing to using. Nutrients do not only provide fuel, they act like signals that tell cells what kind of day it is. When nutrients are arriving, cells tend to favor building and storing. When nutrients are not arriving, cells tend to favor conserving, recycling, and repairing. Neither state is good on its own. The design relies on alternation. That alternation was not invented in laboratories or modern wellness culture. It was forged in a world where food came and went. For roughly 200,000 years, human groups lived in environments that enforced pauses, long walks between resources, failed hunts, seasonal gaps, and the simple fact that night made procurement difficult. In that setting, it was normal for nutrient-driven signals to rise and then fall, giving tissues a predictable chance to shift into quieter maintenance. Inside cells, maintenance is not a single switch, but a collection of small actions that keep order. Proteins that are misfolded are broken down. Damaged parts of the cell are dismantled and reused. Energy production systems are adjusted. In a body shaped by irregular supply, these actions had room to breathe because the environment created time without intake. Even when early humans were not starving, they often experienced meaningful intervals in which the body had to rely more on stored energy. Those intervals mattered because they changed the chemical background inside tissues. The body did not remain in a constant fed signal state. It moved. The idea of constant food changes that movement. If eating is spread across most of the waking day, the hormonal and nutrient signals that favor building can be triggered again and again, while the quieter intervals become shorter and shallower. That does not mean maintenance vanishes. Cells must maintain themselves continuously to stay alive, but timing can change emphasis. A system designed for stronger alternation may, under constant input, spend more time in a mode that assumes more building and less extended quiet. Over years, this matters because aging is, in part, the gradual outcome of how well maintenance keeps pace with wear. Aging is not a moral failure, and not a single mechanism. It is the slow accumulation of tiny imbalances across tissues, small errors in proteins, minor injuries in structures, gradual shifts in how energy is managed. In ancestral conditions, those imbalances accumulated too, but they accumulated under a life that contained enforced variability. Seasons shifted the diet. As a result, activity demanded long intervals between eating opportunities. Night brought a long pause that was difficult to avoid. Even after agriculture began, daily life still had built-in gaps because meals required preparation and work schedules had boundaries. In many older societies, scarcity also returned periodically through drought, disease, or failed harvests. Constancy, in the sense of easy access to calories at nearly any hour, is extremely recent. That matters because it creates a new pairing. Long lifespan combined with an environment that can keep nutrient signals elevated more often than the body historically experienced. A person now may live 70 or 80 years in a setting where the pause portion of the cycle is optional rather than unavoidable. That is not how the human organism was tested for most of its history. Natural selection mostly cared about survival and reproduction earlier in life. It did not strongly sculpt the body for decades of uninterrupted abundance after the reproductive years. So the cellular rhythms we carry may be well matched to a life with genuine pauses, and less matched to a life where pauses are constantly interrupted. This does not claim a single outcome for everyone. Genetics, activity, sleep, stress, and social conditions all influence how aging unfolds. It simply returns us to the central question in a deeper register. If the human body was formed in a world where food arrived in waves, then its cells, like its hormones, may still be expecting waves. When the waves flatten into a steady tide, the body does not become a different species. It keeps running ancient patterns inside new circumstances. And so the real issue is not whether constant food is convenient, but what it means to carry a body built for alternation while living in continuity. From here, the next step is to gather the threads without rushing to a final answer, and to ask what kind of relationship to hunger, pause, and rhythm might fit a creature whose history was never truly constant. When we look back across the full span of human history, a pattern becomes unmistakable. The human body did not emerge in a world of steady supply. It emerged in a world of intervals. For roughly 200,000 years, Homo sapiens lived inside ecosystems that imposed rhythm. Wet and dry seasons, migration and stillness, success and failure, feast and pause, These alternating conditions shape not only behavior, but metabolism, hormones, storage systems, and cellular timing. To understand why the human body was never designed for constant food, we must gather all of these layers into a single view and see the consistency that runs through them. In the earliest environments of East Africa, food was uncertain. Archaeological evidence shows concentrated butchery sites rather than smooth daily accumulation. Plant remains appear seasonally. Mobility was common because resources shifted. This ecological variability ensured that intake fluctuated. Hunger was not rare, and neither was abundance. The body that survived was the body that could store efficiently when food was available and release steadily when it was not. Fat tissue became insurance. Hormones learned to rise and fall in response to irregular supply. Insulin increased during meals and declined between them, allowing stored energy to sustain function. These oscillations were not occasional disruptions. They were the background of life. As humans spread across continents, the pattern remained. Different climates introduced different rhythms, but few eliminated variability. Cold seasons reduced plant access. Herd migrations changed opportunity. Even coastal communities faced tidal and seasonal shifts. The metabolic architecture of our species was refined in this context. It assumed that abundance would not last forever and that pause would return. The endocrine system, energy storage mechanisms, and cellular maintenance pathways evolved within this expectation of alternation. Agriculture, beginning roughly 10 to 12,000 years ago, softened unpredictability but did not erase it. Crops followed seasons. Harvests brought abundance. Yet winter or drought brought scarcity. Storage improved, but spoilage and failure remained common. Meals were structured by daylight and labor. Night imposed a natural fasting window. The ancient oscillation narrowed but did not disappear. Even in early cities with granaries and ration systems, famine remained a recurring threat. The body still experienced meaningful intervals without intake. Industrialization and global trade transformed supply chains and reduced ecological constraints. Refrigeration extended shelf life. Markets operated daily. Eventually, packaged and processed foods made eating possible with little effort. In many modern societies, food can now be accessed at nearly any hour. The structural barriers that once enforced pause have weakened. Yet the biological systems inside us were not redesigned during this brief historical window. Evolution moves slowly. The metabolic and hormonal architecture forged across millennia of fluctuation remains intact. At the cellular level, the same logic appears. Nutrient availability activates growth pathways. Reduced intake allows maintenance and recycling processes to become more prominent. This alternation supports balance over time. When intake becomes frequent and pauses shorten, the timing of these internal states changes. The body continues to operate according to its inherited design, but the environment no longer guarantees the rhythm that shaped that design. Demographic change amplifies this divergence. For most of human history, average lifespan was shorter, and prolonged exposure to stable abundance was rare. Today, many individuals live for decades under steady caloric availability. The body now experiences compressed oscillations across extended time. This combination is historically new. It does not condemn modern life, nor does it idealize the past. It simply reveals contrast. Across environment, archaeology, physiology, and demography, one theme persists. The human organism was shaped within cycles. It was not calibrated for uninterrupted intake across every waking hour and every decade of life. Constant food is a recent possibility layered onto an ancient system. The body still expects variation, still tolerates delay, still carries mechanisms built for feast and pause. If rhythm is woven into our design at every level, then the question that remains is quiet and open. What does it mean to live well inside a body that was shaped by oscillation, in a world that increasingly removes it? If the long record of archaeology, climate, and biology teaches anything, it is that the human body is not a container meant to be topped off all day. It is a system built around change. For most of our species' existence, food was not a steady background. It was an event that arrived, faded, and returned. That repeated pattern shaped the way hunger feels, the way fat is stored, the way hormones rise and fall, and the way cells divide their attention between building and maintenance. The modern world in many places has introduced a new condition. Food can be present almost all the time, and eating can happen with almost no friction. This is not a moral failure, and it is not a simple story of good and bad. It is, above all, a story about context. The body running inside modern life still carries assumptions forged when constancy was rare. When a person eats, insulin and related signals move the body towards storage and construction. When eating pauses, insulin falls, stored energy becomes easier to access, and the internal chemistry shifts toward a different balance. These transitions are not unusual features that appear only in emergencies. They are ordinary functions that make sense only if pause is a normal part of life. In the deep past, pause was enforced by distance, season, effort, and darkness. Even in farming societies, pause was preserved by work rhythms, limited preservation, and the unavoidable gap of night. Only recently has an environment emerged where pause can be shortened again and again, sometimes until it is barely noticeable. In that setting, hunger also changes meaning. The first small signal can be answered immediately, not after a long walk, not after hours of searching, but within minutes. The body begins a cycle, and the cycle is interrupted. This does not mean the body breaks instantly. It means the body expresses less of the old rhythm that once unfolded naturally. Over time, habits adapt faster than biology. Appetite becomes linked to cues of place, stress, and routine, as much as to genuine depletion, because the environment teaches that the signal should always be resolved quickly. Meanwhile, fat storage mechanisms continue doing what they were shaped to do: treat surplus as valuable and protect reserves against future uncertainty. In a world where uncertainty is reduced, that protection may look excessive, but it is still the same ancient logic. At the cellular level, timing matters as well. Nutrient-rich signals encourage growth, synthesis, and building. Quieter intervals help shift emphasis toward recycling worn parts and maintaining order. Cells do both all the time, but the balance can tilt depending on how often the body stays in a fed state. The point is not to claim a single unavoidable outcome for every person. Genetics differ, activity differs, sleep differs, stress differs. Social conditions differ. The point is to remember that the body was shaped under repeated oscillation, and that oscillation was present not only within days, but across seasons and across lifetimes. Modern life has combined two rare conditions: long lifespan and stable, frequent access to calorie-dense food. For much of evolutionary time, few people experienced decades of abundance after the years of reproduction, and even fewer experienced it with minimal daily pause. That combination is historically new, which means the body has not been filtered by selection to be perfectly matched to it. Yet none of this requires panic, guilt, or rigid rules. It asks for clarity. A body built for waves may still function in a steady tide, but it may not feel the same, and it may not distribute its internal labor in the same way. When we notice that, we begin to see constant food not as a simple comfort, but as an environmental condition with consequences that can unfold quietly over years, rather than dramatically overnight. This leaves a final step that is more reflective than technical. If rhythm is part of the design, what kind of relationship to hunger, pause, and nourishment fits a creature whose history was never truly constant, and what might be lost when the last small intervals disappear from ordinary life? If we return to the beginning, to that long stretch of time before farms, before cities, before refrigeration and artificial light, a simple image remains. A small group moves across open land. Some days bring success and shared abundance. Other days pass quietly, sustained by what was stored before. No one expects constant fullness. No one assumes that hunger is a malfunction. It is part of the landscape. The body formed in that landscape learned a language of alternation. It learned that growth and rest belong together, that storage and release must follow one another, that effort and pause are not opposites but partners. Across roughly 200,000 years, this language became embedded in hormones, tissues, and cells. The human organism became fluent in rhythm. When agriculture appeared, rhythm softened but endured. When cities rose, rhythm adjusted but did not vanish. When industry expanded and global trade intensified, The external constraints that once enforced pause weakened. In the last century, in many parts of the world, food became nearly constant in availability. For the first time in our species' history, it became possible to eat from waking until sleep without meaningful interruption. Yet the body that lives in this environment is still the same body shaped under oscillation. It still stores as if scarcity might return. It still releases energy calmly when intake pauses. It still shifts cellular emphasis when nutrient signals rise and fall. It still carries assumptions formed when the world moved in cycles. This does not require rejection of modern abundance. It does not demand rigid rules or extreme restraint. It invites a quieter awareness. If hunger was once a guide rather than a threat, perhaps it need not always be feared. If storage was once protection rather than failure, perhaps it can be understood in context. If cellular processes evolved with intervals of lower nutrient signaling, perhaps those intervals have meaning beyond inconvenience. The body is not confused. It is consistent. It is responding to signals as it was designed to respond, even when the environment that delivers those signals has changed more rapidly than evolution can follow. The central question, then, is not whether constant food is possible. It clearly is. The question is whether constancy aligns with the deeper architecture of a species formed in variability. We are creatures whose physiology was refined by alternating states, whose survival depended on moving smoothly between fullness and hunger, effort and rest, abundance and pause. In a world that increasingly smooths away interruption, what becomes of a body that was shaped by it? And if rhythm is part of our biological inheritance, how might we choose to live so that the quiet language of rise and fall is not entirely forgotten?